This, you know what, and this, your person might want to sit up here. They may want to all sit down there. And we'll see.
All right, so we're getting organized to get started, so if you could come in and take your seats, that would be great, so we could get started on time. Thank you. How do I turn this off?
Here with us, or do you want some?
This number is out of service now.
Good morning.
Hi, good morning, Antonetta. Can you hear us?
Yes, I can hear you.
Okay, great. We're just getting the meeting started. We're having the audience be seated and bringing in the panel. So we'll get started in just a few minutes.
Excellent. And I'm watching it on the website link that you sent me.
Okay, thanks.
All right. Thank you.
So while we're waiting to get started, I'm going to make a couple of required announcements. The first one is the evacuation announcement. Please look around now and identify 2 exits closest to you. In some cases, an exit may be behind you. In the event of a fire alarm, we are required to evacuate this room. Please take your valuables with you and do not use the elevators. While staff will endeavor to assist you to the nearest exit, you should also know that you may find an exit door by following the ceiling-mounted exit signs. Evacuees will exit down the stairways and possibly to a relocation site across the street. If you cannot use stairs, you will be directed to a protective vestibule inside a stairwell. Should we have to relocate out of the building, please obey all traffic signals and exercise caution crossing the street. We don't want you to avoid the falling ceiling and getting hit by a car on the crossing the street. So housekeeping announcements for this room. We have a few housekeeping details to go over. The drinking fountain and restrooms are located out the doors at the back of the room. Go to the left, then past the glass sculpture hanging from the ceiling. The drinking fountain and restrooms are located there on the right. Food service is available in the building on the first floor. Take the grand stairs to the first floor, and at the bottom of the stairs, stay to the right. The cafe is located across the lobby from the security desk. As you might imagine, we also strongly encourage recycling efforts in this building. Please look for the green and tan 3-in-1 containers located outside the doors at the back of the room to recycle your papers, cans, and bottles. Okay, those are the required announcements. So as we get settled, looks like we'll be able to get started exactly on time. I'd just like to say thank you for coming. My job today is to keep the day on schedule. And just to remind you, the main purpose of this meeting is so that the panel members who are sitting up front can have an opportunity to hear the presentations and interact with the presenters. And there will be an opportunity for public participation once the panel members have their opportunity to ask questions, interact with the presenters. And if you have picked up an agenda, you will see that there is some time set aside at the end of the day for more open Q&A. Okay, so we'll get started formally in just about 5 minutes. Well, let's get started. We'll get started 2 minutes early, and I'm sure we'll lose that somewhere down the line. So what I'd like to do is introduce Janice Cook, and she's the staffer from the Central Valley Water Board, who is, along with Christine Job here, has put this workshop together. And if you could maybe welcome people and introduce the panel and kick us off, that would be great.
All right. Welcome, everyone. Thank you for being here. I know it was a hard decision. Did you want to be here? Do you want to be next door in Flows? But they're both being recorded, so hopefully people can get their fill of Delta all day today. I want to acknowledge the support that I had in putting this together. The Delta Nutrient Research Plan has a very active stakeholder and technical advisory group. Many of you are in the room. We really appreciate all of that support because that's making the water board's process better. Particularly a workshop planning subcommittee met for many meetings to put this together, and I extend my sincere thanks to those folks. We have, and I'm going to let the expert panel introduce themselves. I didn't prepare bios, so I'll let you say a few words, each of you say a few words of introduction, and then I just have a brief set of intro slides to remind us all of some of the charge questions. So, all right.
My name is Amelia, aka Amy Ward. I'm Professor Emeritus from the University of Alabama, where I served on the faculty for over 30 years, and about 19 of those years I served as Director of the Center for Freshwater Studies. My general areas of expertise are in inland water ecology, especially microbial ecology in lakes, streams, rivers, and wetlands.
I'm Paul Harrison. I'm from University of British Columbia, Department of Ocean Sciences. My background is on nutrients and algal blooms. I spent 9 years in Hong Kong looking at a similar situation here where we have high nutrients fueling algal blooms. So I'm a Professor Emeritus now. Thank you.
Hi, I'm Hans Pearl. I'm not a Professor Emeritus yet, although people are asking me all the time. I am at the University of North Carolina, Chapel Hill's Institute of Marine Sciences, and my interests span the freshwater to marine continuum with regard to nutrient loads, nutrient dynamics, and their impacts on water quality, particularly at the base of the food web. I've worked in lots of places, and actually my first real job was at UC Davis as a graduate student with Charles Goldman working on Lake Tahoe, and that's where I did my PhD. And I've kind of, I like to distinguish myself from others by having gone the full trophic spectrum from a lake that has a Secchi depth reading of 35 meters to now lakes in China that have a Secchi depth reading of about 3 centimeters. So my interests are pretty broad, but they overlap a lot between physical, chemical forcing features and phytoplankton responses, particularly bloom species.
And one of our panel members could not be here with us today. Family situation meant she was unable to travel. That's Antonietta Quigg from Texas A&M Galveston. And Antonietta, if you're on the phone, do you want to introduce yourself?
Sure, thank you. I'm a professor of marine biology and oceanography at Texas A&M. I have a broad interest in phytoplankton as a bioindicator of environmental stress. I work in estuaries all the way out to the blue ocean, but most of the work is done in the coastal areas. I'm interested in how nutrients impact phytoplankton activity, especially harmful algal blooms. I'm interested in determining how much freshwater inflows our estuaries need to be sustainable and healthy, all the way to working out in the dead zone in the Gulf of Mexico, and what are the different physical, biological, and chemical factors that are driving that. And also doing a lot of work on oil spills and using dispersants to understand the impact on cleaning up the oil versus the impact on the environment. So I'm looking forward to doing this. It's going to be a very interesting workshop. Thank you.
Okay, thank you. So I know you're not here to hear me talk, so I'm just going to give a brief introduction. This is not the first time that we've, folks in the Delta have gathered to talk about nutrients. Let's see if I can do this. Yes. In fact, both Hans and Amy were here in 2009. There was an ammonia summit, framework for researching the role of ammonia and ammonium in the Sacramento-San Joaquin Delta. I'll be talking about some of those research recommendations that they gave us at that time and asking whether we fulfilled them. Other reports that are of interest on this topic, the National Academy of Sciences in 2012 put out a report looking at nutrients and other factors in the Delta. Oops. The nutrients were mentioned in the 2014 report, that, a panel that the Delta Science Program convened to look at Delta outflows and related stressors. And this workshop is being sponsored jointly with the Delta's nutrient science program, but also the Bay's nutrient management strategy. This is just a cover from their current report, but on the reading list for the workshop, Dave Sen's group has already put out a lot of information on nutrients in the Delta, and we want to build on those. So this won't be the last topic, but we really want to dig into some of the questions that are remaining. So going back to that framework report of 2009, they gave, I think, 12 research recommendations, 12 research topics. Research topic 1 was integrative data analyses and modeling. That shouldn't surprise anybody. They recommended that we incorporate hydrology, biogeochemistry, and food web dynamics, and evaluate the role of grazing. We're going to be hearing more about each of those today. We're not there yet, so modeling remains an important topic. Research topic 2 was to look at the sources and fates of nitrogen and phosphorus. A lot of that work has been done. Back in 2009, they suggested researchers here look at longitudinal transects of nitrogen concentrations. We're going to hear some information about that today, using stable isotopes, high-frequency measurements. USGS has picked up on that one, and direct process measurements. And the research topic 4 was links between nutrients processing and phytoplankton populations, particularly rate processes, and looking at measures that could allow us to identify different phytoplankton groups. We've come a long way since 2009. I want to let the panel know that, but we're not done yet. And again, a lot of these topics you'll hear about them in today's talks. Okay, so switching to today's goals. There was a charge for this nutrient forms and ratios workshop and the white paper that's going to come out of it. The stakeholder and technical advisory committee worked together on those charge questions, and I just wanted to put them up here on the slides to remind people. The full charge is on the Central Valley Water Board's website. There are 6 detailed questions that we hope that we anticipate that the white paper will be delving into answers for us. First, can differences in experimental conditions explain differing results from different phytoplankton culture and field studies? Second, what laboratory mesocosms and field studies experiments would help us resolve any remaining uncertainties? When we in the Delta community are looking at designing experiments, what factors should we be considering? And we've asked the panel to help us with experimental design questions, temporal scales, seasons and locations, culture conditions, what actually should the conditions be that we're testing, and what modeling should we be doing beforehand to inform our experimental design? And the last 3 questions, looking forward to the future, What monitoring, special studies, and modeling are needed to track the effects of future changes? That's not a surprising one. What's the range of possible outcomes for the nutrient management in the Delta? And this makes good use, I think, of the expertise of our panel. How does the estuary compare with other nutrient-enriched ecosystems? And with that, I'm going to turn it over to Dave. Are you ready? Our first speaker.
We're going to load Dave's presentation at the moment, so the screens will go blank if you're watching the webcast.
Good morning. Okay, so just right. And
laser pointer.
is there a pointer just for point of reference for Dave? And
It says send. Yes, thanks.
I'd like to introduce Dave San. He's the lead scientist for the San Francisco Bay's nutrient management strategy, and he's a researcher at the San Francisco Estuary Institute.
Okay, well, thank you everyone for the opportunity to present today. I also wanted to thank the staff from the Regional Board in Central Valley for all their efforts organizing this meeting, as well as the stakeholders who participated in that effort. I was asked to give an overview of status and trends of nutrients and phytoplankton biomass in the Delta. As Janice mentioned, most of my work and most of our group's work is focused in San Francisco Bay, so west of the confluence, but we've also obviously needed to reach up into the Delta to understand the loads that are entering San Francisco Bay, and our work is continuing there as well. So I'm going to provide a brief overview of the system and background, and then get into nutrient loads, transformations, changes over time, changes in phytoplankton biomass and productivity, and then the end.
Can I give you a better microphone?
Sure. Thanks. Is that clear? Okay. And then last, I was going to close with a bit of a discussion of changes in the phytoplankton community. You're asking the question, has there been a change in the phytoplankton? What did it look like? So first of all, this is a graphic of how we've been thinking about it in San Francisco Bay, but it translates very nicely to the Delta as well. We have large anthropogenic nutrient loads of nitrogen and phosphorus on the left-hand side, and then we have these beneficial uses or conditions that we're trying to maintain in the Bay and the Delta. And there are multiple ways in which nutrients could have an adverse impact. One of them would be the more classic example of high nutrient loads leading to increased phytoplankton biomass, low dissolved oxygen, and then having various impacts on the beneficial uses, including fisheries and habitat quality. Those same high nutrient concentrations, one of the ideas that we've been talking about, at least in the Bay, is that the high tides lift all boats, and so the increased likelihood, or the likelihood of harmful algal blooms will increase as blooms generally tend to increase, or as biomass tends to increase. Another hypothesis, and one of the reasons that we're here today, is to discuss the hypothesize of the ammonium paradox, or the idea that elevated ammonium concentrations actually lead to lower phytoplankton production and adverse impacts on fisheries, including fish quality, including food for fish. And lastly, the second hypothesis that we're here discussing a lot today is the ecological stoichiometry hypothesis, where elevated N/P ratios or elevated nutrient concentrations could alter phytoplankton communities, either transitioning to harmful algal blooms and toxins or serving as a poor food resource. Another issue that's of substantial importance in the Delta, but not necessarily in the Bay, is the high nutrient concentrations potentially creating good conditions for invasive macrophytes to grow. One of the things I wanted to point out in this graphic is the solid lines indicating pretty well-established mechanistic linkages from nutrients to adverse impacts along that path. And the dashed lines are meant to indicate hypotheses that are hypothesized mechanistic links of which we're uncertain or are not yet well established. So the focus of what I'm going to present, or what we're going to be discussing over the next couple of days, is more around this bottom half of the screen. But we also know that this bottom half of the screen and this nutrient cycling is one part of a much broader set of processes in the system. There's high light levels, turbulence and mixing, grazers in particular in this system, invasive species, habitat alterations, flow withdrawals, contaminants and pesticides, and all of those things also act along some of those direct pathways, modulating the effects of nutrients, as well as acting directly on things like habitat. So we need to be keeping these in mind as well as we're thinking about the nutrient-related pathways. The questions that we have, or I should say that coming from the Bay, I would say one of the main goals, and these are paraphrase some of the charge to the panel. Our goal for the Bay Area nutrient management strategy is to inform next steps related to the ammonia paradox and ecological stoichiometry hypothesis. What are the areas of scientific agreement? If I could boil it down to say the minimum, what are the areas of scientific agreement? What are the specific areas of disagreement, specific mechanisms, specific pathways, and recommendations for next steps? What hypothesis testing or what mechanisms need to be quantified? That would, I think, be really valuable for our effort in the Bay to help us continue planning our science efforts. Although the majority of the heavy lifting of getting this workshop organized fell to Janice and Region 5 staff, on the Bay side, or the Bay effort, one of the things we've been doing is pulling together a number of documents and reports related to synthesizing what we already know. One of those first reports came out in 2014, the Sassoon Synthesis 1. It has several sections that I think are relevant for the panel discussion. A lit review of what people are finding elsewhere about phytoplankton utilization and growth on different nitrogen substrates. A review of the ammonium inhibition studies, nutrient loads, concentrations, and cycling in Sassoon Bay. And then on the right-hand side, this newer document that we just released earlier this month. We tried to pull together things in a slightly different way, realizing that there's a lot of disagreement about the hypotheses. We decided that we wanted to pull together a number of different viewpoints and put them all under one cover. And so we asked Pat Glibbert to write a section summarizing her work, and Dugdale and others to summarize their work. Another lit review about what is being observed elsewhere in other estuaries. A review of the phytoplankton community shift and the data quality. And then as well exploring nutrients versus multi-stressor hypotheses. And so those are all available as part of the meeting materials. As a bit of introduction to the system, I think most of the people know a lot of this or all of this. For the panelists who some of this might be new to them, I wanted to walk through a few things. First of all, this is showing, the red dots on this map are showing DWR, Department of Water Resources Environmental Monitoring Program, long-term discrete sampling stations. These are ones that have been active since 1975 through present. There are others part of this network that were discontinued in 1995. Some other important features of the system are the export pumps located down here towards the south of the delta. It's really important to understand the flows from the Sacramento River and how those enter the system, that those are the largest flows entering the system. They also tend to migrate down into the interior delta, especially later in the season after flows have, as freshwater inputs start to decrease from the San Joaquin. The San Joaquin River enters from the south, and as far as I understand, it disproportionately contributes to the water that enters the export pumps, but don't quote me on that. That's not a hydrologist. But and there's also other really important pieces here are wastewater treatment plant nutrient point sources, the largest of which is the Sac regional plant to the north, but also a couple smaller interior plants interior to the delta, and then a few downstream in Sassoon Bay. And lastly, agricultural inputs from both coming along the rivers as well as interior, which the interior are very difficult to quantify, and that's something we haven't quite gotten a handle on yet. And lastly, something that I believe you'll be hearing about later on today. On this map, this is the Yolo bypass. When flows on the Sacramento River get high enough, flows are shunted down the Yolo bypass, and this cache slough area is an area that recently has shown to be highly productive and may be a source of food for fish. A bit of information on these DWR environmental monitoring program stations. These are, again, monthly samples collected for a full range of nutrients. These are mandated by regulations related to the water extraction from the system. Most of the parameters we would need for basic understanding of phytoplankton and nutrient biogeochemistry are being collected here. I want to point out that they're also collecting phytoplankton community. There was some of the data quality prior to 2007 has some issues that I'll discuss later on. But after 2007, this information, they switched to a method that should be robust. So coming back to now the outline, nutrients, loads, transformations, and changes over time. One of the things we were really interested in is what are the spatial, seasonal, and long-term trends of nutrient concentrations? Based on looking at the data in the system and statistical modeling, what can we infer about the dominant processes controlling nutrient fate? We know that ultimately we need to move towards dynamic numerical models to understand this better, and that's something that we're in the process of developing and I can talk about later, as well as quantify the delta's capacity to transform nitrogen or remove nitrogen. So this graphic may take a second for me to explain. On the bottom, x-axis is showing months from January through December. On the y-axis is dissolved inorganic nitrogen loads coming into the delta in total. And then the key here indicates the darker of the colors is nitrogen in the form of nitrate. The lighter of the color for each pair is nitrogen in the form of ammonium. And so we can see here that this is the Sacramento loads coming along the Sacramento River, entering up here at the northeast part of the delta, northeast corner of the delta, the San Joaquin loads, the loads coming from the east, and wastewater treatment plant loads interior to the delta. And to just give you, these are, I have to say, very rough estimates. They're based on what we think is a reasonable approach to estimating it, based on day flow estimates of water entering and leaving the system, as well as concentrations on a monthly basis at the stations nearby those day flow estimates. So we think a reasonable first approximation. One of the things you can see here is the strong seasonality in the nitrogen loads to the system. You can also see that as part of that strong seasonality, the importance of the sources differ tremendously. But one of the things that's really important to remember as well is that in this upper portion, in the Sacramento-influenced area of the delta, they don't necessarily see the loads that are coming in, or that part of the ecosystem doesn't necessarily see the loads that are coming in from the San Joaquin. So it's not as though these loads enter and then are well mixed and everything is equal or considered equal. One of the things that many of you have probably seen is this paper from JASB documenting the increases of ammonium loads from regional sand over the period 1985 to 2005. This trend more or less flattens off after 2005. And just to give you an idea, this 450 tons per month translates more or less into 15,000 kilograms per day. So one of the things that's really, another thing that's really important for us to be considering is that there are major changes ahead, both in total nitrogen and form entering the system. What I've outlined here in these boxes, these Sacramento ammonium loads, that's more or less equal to 15,000 kilograms per day. And gets smaller in the summertime as some of that ammonium load is transformed most likely into nitrate by nitrification or taken up by algae. But this load is going to be converted all to nitrate, and then it's also, the total nitrogen is going to decrease by roughly 1/3 to 50% over the long haul. And so there's going to be a dramatic decrease in loads, and that's something that Cliff Dahm, I believe, is planning on talking about later on today. It's important as well to understand that we know that the system's machinery is also changing, meaning the things that transform nutrients or prohibit nutrients from being transformed into phytoplankton. We know that grazers change tremendously in space and time and season. We know that light levels are increasing in parts of the delta because suspended sediment concentrations have dropped dramatically. there's indications that temperatures are increasing, or at least will be increasing in the future. And so how do we think about all these things together? A second part of what we wanted to talk about today, or I wanted to talk about, is DIN change over time. So what causes changes in nutrients over time? We have these really, this is from a station in Grizzly Bay in Sassoon Bay, station B7. This is showing dissolved inorganic nitrogen, so nitrate plus ammonium, in micromolar units. And you can see these lots of wiggles, some of those repeatable, maybe, if you were able to extract that sort of seasonal cycle from this. What causes these changes, though, and can we anticipate how future management actions will play out? We know that there's going to be a seasonal cycle to these. We know that there are long-term trends, or there might be long-term trends, some of them that could be monotonic, others that could be increasing or decreasing. We know that the source of water in the delta can change in time and by season. And there's also climate variability that introduces things like, for example, some of these lower lows here. That's happening during the wet season in 2006, a major rain event, and local factors as well. So to give you an idea of the nutrient concentrations, we talked before about how there's large nutrient loads in the system. This is an indication of ammonium concentrations at four stations in the delta. This station C3, right downstream of the Sacramento wastewater treatment plant. C10, down water entering from the San Joaquin. D28A is in the central delta here, and D4. And one of the things that jumps out immediately is the strong seasonal cycle in ammonium concentrations in these other stations, except for the one just downstream of Sacramento's wastewater treatment plant. Most likely the flat line here is because of the short travel time between the discharge and where measurements are being taken. But something that's really interesting, I think, here is that if you look at what the composition of the water is at D28, a large portion of that water is actually Sacramento water, Sacramento River water. But it has, first of all, lower concentrations, and I apologize, I'm jumping back and forth between units a bit here. These are in micrograms N per liter. You can see at this more interior delta station, even though most of the water there, or much of the water, is from the Sacramento River, that it does experience this long, or this pronounced seasonality. And one of the reasons for that, we believe, is that the travel time from here to here is more or less on the order of 20 to 30 days. So there's plenty of time for transformations to take place and for nutrients either to be taken up or decreased. I just got the 3-minute warning, so I'm going to speed it up a bit. All of this material is part of the materials that were provided to the panel. So here's showing nitrate concentrations and dissolved inorganic nitrogen concentrations. Again, seeing some of the same things that here, it's not, for ammonium, it's not just conversion of ammonium to nitrate, but there's actually either that nitrogen is being pulled into the organic pool or it's undergoing denitrification in the system. I know some of the panelists were interested in total nitrogen concentrations, and just to flash those up here as well. What are the dominant processes controlling nitrogen concentration in fate? This is showing ammonium concentrations in milligrams per liter, and showing the strong seasonality only from the period 2000 to 2012. And one of the things we wanted to do here, that you can see in here that there's a lot of similarity in these patterns, but the patterns are, but there's also different baselines, and there's also some departures from a typical trend. So we used an approach called empirical orthogonal functions, which is a PCA, or principal component analysis, time series analysis. And we looked at what the different principal components are that can explain the ammonium concentrations. And it turns out that you can explain something along the lines of 70 to 80% of the variability in the ammonium concentrations using just 2 time series of these empirical orthogonal functions. And we think that what's causing these transformations, a lot of these peaks here are occurring during the cooler periods of time before freshwater flows have increased. So a decrease in nitrification rates and uptake rates, and an increase in what you're seeing at a downstream station. This is a signal of flow and dilution. And then lastly, right after that is a period of increased temperature where that's increased utilization. So we think that there's a lot we can learn from this data, especially looking across multiple sites. One of the other things I was asked to talk about is fate and transport, or loads. And this is also summarized in one of our recent reports. We did a summer whole delta mass balance from 2006 to 2011, estimating the loads entering and leaving the system. We did it at a whole summer because you can't do instantaneous upstream and instantaneous downstream and look at the transformations in between because of the relatively long residence time in the system. So this is definitely a sloppy average, but it's something that gives you a good indication. So we estimated that around 25% of the ammonium that, or the total nitrogen that enters the delta is removed either by burial or denitrification or uptake by macrophytes, for example. And ammonium, on the other hand, it's 65%, a large transformation of ammonium as it moves through the system before it enters Sassoon Bay. I'm going to jump ahead to one other quick thing. I was asked to cover changes in phytoplankton biomass. This is something that I think almost everyone here is quite familiar with. And this is a graphic showing combination of USGS and the DWR EMP data in Sassoon Bay, the substantial loss of phytoplankton biomass after 1986, and this is one of the idea that one of the thoughts is that this is contributing substantially to adverse impacts on pelagic organisms. In the Delta, I won't present any of this, but a really nice review of this is by Jasby 2008 and Jasby 2000 et al. 2002, showing that there's a general decrease in chlorophyll in the Delta as well from 1975 to 1985, but that it lags behind Sassoon Bay. And in addition, you can't explain that loss of chlorophyll in the same way in the Delta as you can in Sassoon, because the organism that invaded is more or less a saltwater species. It's not one that tolerates low salinity very well. So the same explanation doesn't stand. Jasby 2008 also identified an increase of over a period of 1995 to 2004. So one of the last things I wanted to cover was a bit of something that surprised me tremendously. When this is showing dissolved inorganic nitrogen concentrations in micromolar this time at a station in Sassoon Bay. And it's broken down into different eras, so you can see the seasonal cycle. One of the things you can see is a pretty sharp increase between 1975 to 1987 and the second and third era in dissolved inorganic nitrogen concentrations. And my first reaction to seeing this several years ago was to assume that this was entirely due to an increase in loads to the system. But what's interesting is that Wim Kimmerer was able to demonstrate, looking only at silica data and increases in silica concentration in Sassoon Bay, that there was a substantial decrease in diatom production. And that you can see here in the second graphic, the jump up in silica concentrations as well during this time period. And I won't show you the more detailed version of this data, but diatoms take up nitrogen and silica in roughly a 1-to-1 proportion. And so this increase in silica from 150 to 200, you could say the same nitrogen could have increased by about the same amount in that same month. 50, the nitrogen concentration should be up around 60 or 70 due to that loss of phytoplankton or diatom production. So one of the things that this really made me think is that the perturbations, the biogeochemical or ecological perturbations to the system, can be of similar magnitude as the changes that you might think from increased loads over time. So Sac Regional's loads of ammonium have increased by a factor of 2, and it's possible that the clam invasion in 1987 could also cause as much as a factor of 2 increase in dissolved inorganic nitrogen concentrations. So with that, I think I'll wrap up, and I can come back to some of those other slides later if there's interest. To summarize, there are large nutrient inputs to the delta, elevated nitrogen and phosphorus concentrations. Those concentrations generally substantially exceed the half saturation constant, so there's not kinetic limitation of phytoplankton growth. And that's one of the big challenges here, is to understand what actually controls phytoplankton community in a system like this when nutrients are generally replete. There's abundant long-term data on nutrients and bulk phytoplankton biomass for us to really, I think, make some major advancements in our understanding of how the system behaves. I'll jump down to my last bullet, though, but ultimately we're going to need coupled hydrodynamic and biogeochemical models to really unravel these multiple intertwined parts. The delta is a vigorous biogeochemical reactor. We're moving in summertime up to 30 30% of total nitrogen and up to 60 to 70% of ammonium before it enters San Francisco Bay. But it's a complex system with numerous factors that influence the observed concentration. So one thing I didn't mention was the trends over time, and I can come back to that if there's interest, but you can see some clear trends of increase over time, but others are difficult to detect because of all of these other fluctuations that happen in the system. There were major changes in chlorophyll throughout the system, both in the Delta and Suisun Bay, with the Delta changes lagging behind Suisun, and it not being realistic to assume that the same mechanism can explain both. There is an invasive clam as well in the Delta, but that clam invaded much earlier than the 1970s, and it still exerts likely a very substantial influence on biomass, but not necessarily that there wasn't an abrupt increase. And that's something that perhaps Jan can correct me on later on today. Lastly, the phytoplankton community data pre-2007 has some pretty severe data quality issues. And although I didn't present it here today, it's part of the documentation that you received. And although we reanalyzed the data not with the goal of challenging directly any of the hypotheses about a shift in phytoplankton community related to nitrogen and that causing impacts up the food web. But when we went in to analyze that data, we did find that there's these really substantial data quality issues that I think make it very challenging to assert what kind of change actually happened, including ones that could have been influenced by nutrients. And that's all. Thank you very much.
So Amy, you guys can have some time to ask him some questions if you'd like.
I'd like to ask hopefully a quick question with regard to the statement about Delta Vigorous Biogeochemical Reactor, that is, it retains and transforms nutrients. If I read your numbers correctly, the system appears to retain 75% of the total nitrogen and exports 25% of what goes in. Is that?
It's the other way around.
Okay,
yes.
So it exports 75%, which is a relatively low retention rate. Obviously, phytoplankton can be one of the players in that in terms of transforming nutrients. Do you have a sense of their importance in comparison to other mechanisms of retention and transformation in the system? Or is it pretty much a black box at this stage?
No, I think that's a great question, and it's something that we're interested in doing, is so Jim Klearn and Alan Jasby have been able to estimate phytoplankton production rates or productivity rates in the Delta. I think, and they've been able to estimate those changes over time. Although there's a lot of uncertainty in the exact nitrogen proportions relative to chlorophyll, I think we could still get a back of the envelope estimate of the change in nitrogen uptake that may have occurred due to changes in productivity over time. So I do think it's possible. And regarding whether 25% of total nitrogen is small or large, I think that given the huge influx of nitrogen, it's still, somewhere on the order of several tens of thousands of kilograms per day. There is pretty substantial, that's nonetheless a substantial removal, especially when a large portion of it is piped along a river channel that isn't exchanging with the flood.
Right. My reason for that was based on some published studies that have been done in watersheds across the country, and typically export of 25 to 30%
would be considered low. Okay.
About the average among
okay.
those.
I guess
Without qualifying it as high or low, but nevertheless, that's about what it is.
Okay. Thanks.
You mentioned the total nitrogen, and then you had up there organic, but there's a quick percentage. What is the range of the organic N of total nitrogen?
It's in the vicinity, it varies a lot by season and by station. It's in the vicinity of 20 to 40% organic nitrogen.
Okay. And second question had to do with bacteria. So bacteria are a big player. Do you have any idea of the bacterial biomass and what kind of role they play in taking up the inorganic nutrients and then processing the organic nutrients into inorganics?
I don't think we have a really good handle on that yet. There are a lot of people who are thinking about issues related to the abundance of food, either through the microbial food loop or the microbial loop or the phytoplankton or detritus. And it's not, I don't know that the microbial or bacterial side of that is well constrained at this point.
But that would be a key component of any models that you're going to do.
Dave, what happened to the light? Can you talk a little bit about what's happened during the course of the time series that you showed? What has happened to light and turbidity in the system?
Well, depending on the era that you're talking about, and the graphic that jumps out of my mind most clearly is one for Sassoon Bay, and it's from the paper by Klern and Jasby. And it shows that suspended particulate matter concentrations have decreased by a factor of 2 from 1975 through roughly 2010. And so if you decrease the suspended sediment concentrations by a factor of 2, you increase the light levels, the other nutrient, the light nutrient that phytoplankton need. And so that is really a dramatic decrease over time, dramatic increase in light levels. One of the things that is really tricky is that when we look at high frequency data, in particular in South Bay, that there's really large fluctuations due to tidal resuspension of sediments. And so I think it's pretty challenging to know exactly what those translate into during the growing periods of the phytoplankton. But nonetheless, have.
Yeah, so presumably that would act to increase the phytoplankton, if anything, during the course of that time. And the other question I had was deals with the lag in the delta for chlorophyll. And I'm curious whether that might be have something to do with the way chlorophyll is measured in the system in terms of, is it an integrated number? Is it surface chlorophyll, deep water, or throughout the water column? And the reason I ask that is because if there has been an increase in surface bloom-forming cyanobacteria in the system, it's possible that some of that chlorophyll could have been missed.
Yep, that's a great point. And as for most of the chlorophyll measurements, I believe they are collected 1 meter, grab samples 1 meter below the surface. And so the cyanos are not taken into account. And as well, I could imagine that if the photic depth is increasing over time, that's one way that productivity could be increasing as well, is that you have a deeper layer of chlorophyll at the same concentration.
Yeah, so I'm just suggesting that possibly, looking at the delta itself, I think it probably is useful to look at how those data were collected.
Yep.
That may be an issue there. And the last question I had was flow-corrected data. You want to comment on that, or
did you see that as
in terms of loads?
Yes. So a slide that I did not show is
the reason I asked that question is because we have very similar. Well, in general, there's a lot of issues revolving around what happens seasonally when you compare non-point sources to point sources, and typically during the summer, the point sources become more important.
Yep.
Just because of the reduction in flow.
So
Dave, can I ask you to hold the answer to that until the end of the day so we can move on? But
okay.
Make a note and please don't forget. Okay? Thank you.
Thank you.
We have a 20-minute break built in, but I think people are going to want that break, so it's good to have the discussion. Okay, our next speaker is Tamara Kraus. She's a scientist with the U.S. Geological Survey. I continue to be impressed by the number of projects you're involved in, but she's with the Biogeochem group, right?
Okay, again, thanks. It's nice to meet everyone and be here today. I am here talking about one specific study that we did on the Sacramento River, where we implemented an effluent hold to the river in conjunction with regional SAN. And I'm hoping if I have time, I can also talk about some other research projects that our group has going on. I want to recognize my colleagues, especially Brian Bergamaschi and Brian Downing, and we'll be hearing from Liz Stumpiner later today.
Yeah, and is it just me or is it?
Okay, try that. How's that? Much better. Okay. Okay, so I put the charge up here, but Janice reviewed that. But really what I see my role today is to tell you a little bit about what we've learned in the last couple of years for the panel so they don't have to read every document and every sentence. I also want to highlight here that I think we all understand that there's multiple controls on phytoplankton, and Davidson gave an introduction to that. There are tons of conceptual models and box plots. These are some of the main drivers that I put up today. Of course, there are others. And the complicated thing is not only are there multiple drivers, but all those drivers interact in complex ways. So we're really focused here on nutrients and also the role of ammonium specifically and the stoichiometry of nitrogen and phosphate and ammonium and nitrate. I'm going to focus mostly on the sources of ammonium from Sac Regional Wastewater Treatment Plant. And again, we can do our best to try and do lab studies and incubations and even look at transect data to understand what the effects of nutrients are. But really what we want to know is what are the effects of nutrients in the natural system at the landscape scale, and whether observations that we observe in mesocosms and controlled studies actually apply and are still basically apply at the landscape scale. So what I'm going to talk about is a landscape scale experiment that we did where we actually halted effluent discharges. There is a paper in press with limnology and oceanography on this, so if you want more information, please refer to that paper. Okay, to give you a background on what the study was, we did this effluent hold in October 2013 and June 2014. We halted effluent to the Sac River for about 20 hours, which resulted in about a 15-kilometer stretch of river that was free of effluent and thus had no ammonium coming from the wastewater treatment plant entering the river. This, we used a Lagrangian approach where we tracked the parcel from about 20 kilometers upstream of the wastewater treatment plant to 50 kilometers below. It gave us about 1 day of travel time to get our baseline of what the different parcels look like above the wastewater treatment plant, and then following the parcel for about 4 days as it flowed past the wastewater treatment plant. It was pretty big field effort. We used various methods to make sure we were actually in the designated parcels with and without effluent, using water velocity measurements at the monitoring stations on the river. We also employed drifters, but the most important thing and one of the great outcomes of this study was that we built and improved our ability to track water quality in real time with a roving boat, able to map water quality. This is the data that, that's the map of all the samples we collected, just to give you an idea. But this is a figure, really, this is fluorescence of dissolved organic matter. We also saw a similar pattern in specific conductants and downstream nitrate that was being nitrified from the ammonium. So we could very clearly see when we were in the wastewater versus the non-wastewater containing pulse of water. Again, Davidson gave you a little bit orientation in the delta, but just to quickly orient you to this study, is we are talking about the channelized section of the Sacramento River. We're up here in Sacramento. We started upstream at the I-80 bridge. You'll see a lot of these sites mentioned in some of the other papers that are on your reading list. So I just wanted to point them out. Here's the I-80 bridge. Here's where the wastewater treatment plant, Sac Regional's outflow is, right here at Freeport. Hood is a DWR long-term station, equivalent to C3 in some studies. And then you go past Walnut Grove, and here at Ileton, you pass Ileton and you enter what's called the cache slough complex. And you'll hear more about that, studies in the cache slough complex from Liz. But to orient you quickly, can you run the animation? Just to get some hydrodynamics in here, a lot of you have seen this visualization that Marion Guerin made for our study. What you're going to see here is this is a particle tracking model that's first releasing particles that are purple in color from this wastewater treatment plant here at Freeport, and you'll soon see a slug of blue water that's representative of about a 12-hour period, in this case, when we ran the simulation of when basically water passing the Sac Regional is tagged with blue particles to represent a slug of water that does not receive effluent. So here it comes in. What I want to have you look at here is that even this reach of the Sacramento River is tidal. This was during our study, there were low enough flows that the river actually reversed even at the wastewater treatment plant. And that after 4 days, you can see that the slug of blue wastewater-free water starts entering what we call the mix master of the delta, where things get very complicated. There's particles can go in lots of different directions, and the residence time of all this different water really is quite variable. And this animation runs for a total of 14 days under the predicted flows that were expected about a week before we actually did the study. Next slide is so. And Liz is going to show this again, so you'll have another chance, I think, to see it. Okay, so one of the reasons we did this study is there were several prior studies that did show that there were documented declines in chlorophyll concentrations along the Sacramento River. So that is something that was well accepted. That when you travel all the way from upstream, about 20 kilometers upstream to 50 kilometers downstream of the wastewater treatment plant, we see this progressive decline in chlorophyll concentrations. This is from Pho et al. 2010, approximately monthly sampling over a year-long period, and we saw they documented declines in chlorophyll along this river reach. The reason I grayed out this area is I think it's important for you to think about when you look at all the different papers. Again, this is the section, the channelized section of the Sacramento River, where you have pretty good ability to watch an intact water parcel moving its way through the system. But as soon as you reach the mixmaster of the delta, you can, it's much harder to really put your finger on what the residence time of that water is, what the source water is, and that water is really sloshing back and forth, so the water's moving underneath you. So when you start pinpointing a location, it gets a little bit messier when you're in the delta where water's really moving around on tidal timescales. Okay, and in addition to the Pho data, there's also a paper by Parker, 2012, 2 papers by Glover et al. In 2014 that came out that also looked along the same stretch of the Sacramento River, starting from above the wastewater treatment plant, going all the way out to Curtin Straits in some cases. And again, I grayed out the area that's below Isleton where the flow gets really complicated, but you can see progressive declines in chlorophyll in green here, and also this black line here. So again, well established that we see declines in phytoplankton on this stretch of the river, but really what these papers looked at not only are the declines, but also what the health of the phytoplankton is. And they saw that not only are you seeing this shift in ammonium, in uptake of nitrate to ammonium as soon as the phytoplankton are passing the wastewater treatment plant when you have high ammonium in the system, you see that expected preferential uptake of ammonium over nitrate, but also they document declines in primary productivity, the FB versus FM, and I think Renee is going to talk about that measurement later, and a change in ratios of phytoplankton to chlorophyll, and also some indication that there's declines in diatoms over other species. Okay, so back to what we did in our study. During the Lagrangian study, we measured most of the things you would expect, like nutrient concentrations and chlorophyll concentration. We also did potential uptake of nitrate and ammonium, as well as carbon, using tracer techniques. We did phytoplankton enumeration as well as visual inspection under the microscope with Kirk Carpenter, who was our phytoplankton ecologist on this study. He's up in Oregon. There was also some parallel enclosure studies done by a master's student working with Alex Parker and Francis Wilkerson. We did zooplankton enumeration, and Regional SAM also conducted a benthic trawl survey so we could get an idea of what losses to grazing were along the stretch of the river. We also looked at stable isotopes of nutrients and particulate organic matter, but I won't talk about that today. Okay, so I want to give you the results of the study, which were, there's just a couple slides really. And that was a complicated study to do. The results are pretty straightforward. We saw, obviously we saw an increase in ammonium as soon as you pass by the wastewater treatment plant. And these slides I'm showing, these slides I'm showing travel time on the x-axis here with negative time being when you're upstream of the wastewater treatment plant, then 0 is the time of travel when you're passing the wastewater treatment plant. So this is the 5 days of travel. You can see that we elevated the ammonia concentrations with the influx of wastewater in these darker filled symbols, and then the light symbols here are the October and June samples for ammonium concentrations that did not receive effluent. Okay, so we have these 2 parcels traveling down the river, one that has high ammonium, one that has low ammonium and no wastewater. And this is what we saw. Really, the point here is that we didn't see any differences between the parcels that received effluent and the parcels that didn't. Either in the chlorophyll concentration, we saw no differences in carbon uptake, and we didn't see any significant differences in the assimilation number, which here is carbon uptake divided by the chlorophyll concentration. This is just a table from the paper with the statistics. I just wanted to point out that, again, we saw a significant change in the concentrations of nutrients, elevated ammonium, with the presence of effluent in the plus effluent parcel. We also saw a higher nitrate going downstream due to nitrification of that ammonium. We saw the expected shift from uptake of nitrate to uptake of ammonium in the presence of effluent, but all the other parameters we looked at as far as what was happening with the phytoplankton itself were not significantly different between the 2 treatments. I stuck in this slide on the species composition. The main takeaway messages that I have here are that we saw a dominance of diatoms in these species. They're mostly in blue colors, so you can see there's a lot of blue there. And the other takeaway point is that these were largely benthic facultative planktonic diatoms that were forming filamentous colonies, kind of ribbons that were able to float down the river. And again, I learned a lot by doing this study from Kirk Carpenter, who's more of the phytoplankton expert on our team. But he found this very, one of the important points he asked me to emphasize again is that these planktonic diatoms, that they weren't really true planktonic diatoms, suggesting that there's some other processes that we have to think about where these diatoms are coming from. Are they coming from the sediment and being entrained in the water column as the water is moving down the river? Where's the habitat really? Where are these diatoms really growing and multiplying? He, in the microscopes, we saw that there was a large decline in chlorophyll concentration, and he did observe downstream declines in the health of the cells. There was also a lot of broken and empty frustules noted by the enumerators, which also was some evidence of material being brought back into the water column from the benthos. As far as doing ordinations on the species composition, again, there was some difference between the 2 different sampling dates, October and June, but we saw no significant differences in species composition between the treatments. And what the other point I wanted to make about the phytoplankton enumeration data is that they were quite heterogeneous, even between several hours between sampling in the different Lagrangian parcels, which also indicates that there were probably other controls on really the samples that we were collecting. These were grab samples at about 1 meter depth. So again, time of day, where we were in the tides might have been affecting what species were actually in the samples we collected. Okay, and then I want to make an important point here that, again, Nicole Travis in her master's thesis presents data from parallel mesocosm containers where she collected water from the river, put them in these mesocosms, and then floated them down the river for 5 days during the study. And this is data from her thesis, which shows that in the absence of effluent, there was really no growth of chlorophyll, but in the presence of effluent, there actually was significant growth by day 4. Chlorophyll concentrations had increased over 40 micrograms per liter, similar to what was seen when she spiked the water with ammonium and nitrate. So the take-home message here is that the mesocosms showed very different results than what we saw in the river. Although we saw phytoplankton declines in the river, in the mesocosms, we actually saw that there was potential for growth if you took that water and put it in a container and floated it down the river And there was some indication that there wasn't growth in the absence of effluent, suggesting that there might have been nutrient limitation, actually without effluent being added in this case, because nutrients were quite low coming down the river at that point. Of course, we have to keep in mind that in these mesocosms, we are not experiencing the same conditions as what's going on in the river, which is really why it's so important to do these landscape scale experiments. So the things that mostly come to mind that are different is that these mesocosms are at the surface, so of course they have much greater potential for growth because they're under high light conditions. They have zooplankton in them, which might actually have an advantage of being able to munch on the phytoplankton because they're all stuck in the mesocosm together. But as far as clams, there are no clams in the mesocosms. And then very importantly, they're hydrologically isolated, so they're not going to experience things like sedimentation or resuspension of other material. Okay, so although I could go on and tell you about some other studies, I know I'm out of time, so I'm just going to try and breeze through and tell you some of the other stuff that our group would like to do as well as that we're working on, so that if you have any questions about that, you can either ask me or our colleagues to elaborate on that. So the first thing is that we learned a bunch of things from this study. I think one of the questions that came out is why are chlorophyll concentrations at times 20 micrograms per liter up at Sacramento? Like, what are the controls that are actually leading to there being higher concentrations up in the river, whereas pretty much we always see in the Sacramento River concentrations of phytoplankton between 1 and 3 micrograms per liter. So even if you do have a bloom happening upstream, it doesn't make its way down the river. And, you know, it's kind of interesting to wonder what's controlling the blooms upstream. Not that they will necessarily ever make it downstream, but it's still interesting to know what allows phytoplankton to reach higher concentrations upstream. And regional sand has started that. Actually, this is my conclusion slide, so I actually thought I was on this slide, but I think I made those other points clear. So regional sand has already started looking farther upstream. We also think that results from this study, do highlight that we need to get a better handle on hydrologic effects on phytoplankton, in particular what the effects maybe of the changing water velocities and turbulence have on the role of these facultatively benthic phytoplankton moving basically from the sediment into the water column. And also, even though a lot of the ways we look at the phytoplankton enumeration, people just classify diatoms as diatoms, it's curious to see whether it's the same kind of diatoms or whether there's actually a shift in the diatom community itself. We think there's some, again, we know that there's nutrients coming in from the wastewater treatment plants, but we don't have a good handle on nutrients coming from the benthos, which we think is also important. We did see some release of nutrients, which we assume is coming from the benthos in the wastewater-free parcel as it traveled down the river. And of course, looking pre and post wastewater treatment plant upgrade. And then I put here kind of the things that were brought up in the 2009 review framework of the ammonia hypothesis again. We think that we have to beef up our monitoring so that we actually have our eyes on the delta so we can see when blooms happen and when they don't and what the conditions are that might be triggering the blooms, and help create data to build these linked biogeochemical physical models, as well as considering intercalibrated approaches between our different studies.
Thanks very much. As we start the questions, I just want to make sure that we have our other panel member, Antionetta, still on the phone. So if you have a question, just please butt in because we can't see you, okay? Are you still there?
Yes, I am.
Great, thank you. Okay, go ahead.
I had a question.
Mulling over.
I've been doing benthic cores and measuring benthic fluxes and sediment oxygen consumption.
Sorry, I missed the first part of that question. Can you repeat that? I understand you asked if people have looked at benthic fluxes.
Benthic nutrient fluxes and sediment oxygen consumption in the same areas that you're making these measurements that you presented today?
So the quick answer to that is I have not done any of those measurements. There are a few papers out there that have looked at sediment release of nutrients. I can think of one USGS report that I could provide if you're interested. And there has been some studies looking at nitrification rates, which of course could, both in the sediment, bacterial composition in the sediment that are nitrifying, which one could think about how much oxygen consumption is happening related to nitrification itself. But I don't think there's a lot of information on that, but we don't have a problem with dissolved oxygen, low dissolved oxygen in this section of the delta.
Okay, thanks.
So what you were finding in terms of phytoplankton composition was predominantly diatoms, and you think from benthic sources based on the taxa. And the mechanism you think for how they get in the overlying water from the benthos, do you have?
Well, you're getting me into slightly uncomfortable. Like I said, I'm a biogeochemist and I try to team up with the phytoplankton folks. So really, Kurt could answer this more clearly, so I'm kind of paraphrasing what I've learned from him. But yeah, that was, again, the fact that the diatoms that were predominant, especially on ulna, Maria ulna, and Synedra, and we've seen also Alacrocyra, and I think also in the deep water ship channel and coming down the tow drain, there's been some mention of this meroplankton type of phytoplankton that are, it just makes you think that you have to think about where are those cells really coming from? Are they able to live in the sediment and then populate the water column when conditions are good? And is that what the long-term data is showing, that those are the populations that have been in the delta?
What were some of the, do you know some of the non-dominant forms, the non-diatom taxa that were there?
I don't want to try and pronounce them off the top of my head, but there's a table in that paper that has all the taxa and their percent by biovolume if you're interested.
Rather than the specific taxa, maybe just the general groups such as cyanobacteria or greens or something?
There was not significant cyanobacteria, so I can put this.
That's okay. I can.
Yeah.
I can look.
So you can see here there were a couple. This is a longer-term data set that Regional San actually collects at Freeport here and then downstream a little bit. And you can see there was a couple of blooms of Spirogyra and Cyclus. I'll let you say that.
And just one last quick question. Given that I think you said there was substantial, when looking under the microscope, that the diatom frustules were broken, and perhaps, do you have any sense of the physiological health of the benthic component that's appearing in the overlying water, or could possibly some of the metabolic processes that you're assessing come from some of the non-diatom taxa rather than what was dominant in numbers or biovolume or whatever?
You're saying, sorry, I'm not sure if I understood that question. You're asking if the
I'm asking specifically about the non-diatom component of the one you just went over, and wondering if the uptake, for example, in other metabolic processes that you're assessing could be due to that group rather than what might appear to be moribund benthic diatoms in the overlying water.
Yeah, I actually think that's very important. It's kind of a confounding factor when you, especially if you're trying to normalize your rates of nitrogen or a nitrate uptake by some parameter that's confounded by the fact that not all of your phytoplankton is live. That's one reason that we use the assimilation number, where we normalize the carbon uptake by the chlorophyll concentration, because mostly if you just collect the phytoplankton on a filter and try and look at total carbon or total nitrogen, you don't know how much of that total carbon or nitrogen pool is actually a lot of phytoplankton. So certainly that's one of the issues, that when you do stuff in the field, you're just looking at a bulk measurement. You don't know who's really the actor, and so then you go back to the lab and try and isolate species, but then again, what you're measuring in your lab-controlled study is really representative of field conditions. So that's why we got to do both.
Thank you.
I have a question on the spring neap tides, and from your model, looks like a lot of sloshing back and forth, and so on the spring tide, this could be the stirring mechanism that brings up these benthic diatoms, and all of a sudden you have this sort of high bloom for some unknown reason, and then in the neap tide, things may be more quiescent. Anyway, do you take into account when you're doing sampling, whether it's a spring or neap tide?
I'm going to throw up this visual, which is a recent visual that Brian Bergamaschi made, just to say that we're trying to do that. So again, this is chlorophyll concentrations at Decker Island for this year. I don't know if anyone has told you that, well, we've tried to all be out in the Delta taking lots of good data to be there when the phytoplankton blooms happen so we can figure out why they're so happy. And 2016 was a good year for that. We saw massive phytoplankton blooms across the entire Delta. And Brian is actually working on that data set, trying to figure out if you look at all the measurements we have, if you look at flows, if you look at temperature, if you look at nutrient concentrations, can we figure out what conditions really did allow this bloom to be triggered and then be sustained through the Delta. And I think actually you kind of hit one of the parameters that's coming to the forefront, which is possibly water depth related to the spring neap tides, and that's just, I think that's a really good question. Is again, what is really allowing phytoplankton to grow and what's preventing them from growing in the Delta?
Okay, one more question.
Oh, that's cool.
Can you just go back to your conclusion? What's your main conclusion about what's really controlling phytoplankton production based on these experiments?
Well,
or based on, the field experiment?
Well, I would say that really the main control on phytoplankton in the Sacramento River that we observed was really loss, not production. So we saw that the factors leading to phytoplankton loss overwhelmed any rates of total growth or production.
But really, you're talking about residence time. Is that right?
Well, so yes, we followed parcels of water as they traveled downstream for 5 days. And really one of the things to consider, I guess I will go back to, this slide is really, you're right when we, it's kind of unsatisfying. We say, well, we looked at phytoplankton, it declined equally in the presence and absence of effluent, which sort of, the next question is, well, why are they declining? And so we did try and look at the available data. We looked at our losses potentially due to zooplankton and grazing based on published rates of losses, and that could not account for our losses. We saw upward of 50% chlorophyll loss in the upstream reaches above the wastewater treatment plant over a 24-hour period. So grazing couldn't explain that. One idea is that, which we think some of the future studies that are being done upstream could answer, is that upstream of our study reach, even before where we started sampling, the river is a little bit shallower. So maybe just the fact that the river is entering a deeper region and the photic zone is effectively changing could have made the phytoplankton less happy. But we also think that hydrodynamics might also play a role, because you're also going from a flowing system to now a system that's becoming increasingly tidal. So that's where this idea that maybe upstream you have more consistent flows, and downstream something suddenly happening to the phytoplankton now that they're experiencing these variable velocities, slack tides, different turbulent conditions that might be affecting their ability to stay in the photic zone and their ability to just
well, your experimental enclosures would indicate that light limitation is probably pretty important, because they were incubated near the surface, right?
That's right, yes.
Yeah.
Okay, we're going to have 20 more minutes built in before lunch to pursue some of these things, so hold on to your questions. Thanks very much.
Sure.
So we're going to take a break now, and we will reconvene at 10:40 at 20 till and start the next presentation. Thanks.
I did not.
Nothing to do with the question, but you have it in there. Access to the presentations.
Hello.
that works. Yep.
Let me see if I can get a cover for you. Let me see if I can pull one of those covers off the microphone. Okay. That way you don't feel like you have some weird metal object just about ready to
Have access to the PowerPoint presentation?
Yeah, Paul was asking, so we'll make sure that you guys have it available to go through the slides. Especially too if you want to ask, because we're kind of short on time, so we're kind of cutting people off with questions. I think that maybe on the second day, if you want to ask some questions you didn't get a chance to ask today to the presenters, you'll have an idea what slides you want to go through.
All right, we have 2 or 3 minutes before we get going again.
Make that green. Sounds good. Oh, for the mic. Yeah.
Okay, we're going to get going in the interest of good governance.
Staying on time.
Okay, Aaron, let's go.
All right, I think we're going to...
Take your seats, please. We're getting started. Just go and they'll...
I'm going to start talking, and you are all going to sit down.
Please, we're starting.
All right. So you'll notice one thing's new. We've switched microphones because apparently the other one wasn't working as well. So now I get to hold this one, and that feels pretty good. All right. So first of all, I want to thank Janice Cook and the Regional Board for organizing this and all the hard work they put into organizing the workshop over the next 2 days. My name's Aaron Strong. I'm an assistant professor in the School of Marine Sciences at the University of Maine. And as much as I'd like to talk about nutrient forms flowing into Casco Bay in Maine and some of the wastewater questions we have there, today I'm going to talk to you about an experiment I did when I was at Stanford University previously, which speaks to many of the questions that are before us today. If you go back to Dave Sen's model in the beginning, conceptual model, we're talking about the hypotheses on the bottom end of that graph today. And that's what the experimental work I'm going to talk about today speaks to, both the question of ammonium suppression of nitrate uptake and thereby suppressing growth in phytoplankton accumulation, and also the question of nutrient forms and their impacts on phytoplankton community composition. So here's an overview of the presentation. I'm going to start with a little introduction to DIN conditions in the lower river, but you've seen many of these graphs today, so I'm not going to belabor those points. I think we're all pretty familiar with them. Then I'll speak to the experimental design and treatments that we used in our incubation study, go through the results, and talk a little bit about what's next. So Focused on the lower Sacramento River here, these are plots of ammonium concentration made with a high-frequency underway ammonium sampling setup. So this is it's a wet chemical setup that works through sampling flow-through systems, and we get samples about every 200 meters or so going up and down the river. And the key point here is that these white data up here are actually data, as you can see here. And no matter the season, we see the same trend, right? This high pulse of ammonium coming from the Sac regional plant. And in May of 2015, we added 2 other underway measurements of nitrate and of FV over FM, which Monet will speak to as well. And so this is just to give you an orientation to some of the questions we're talking about before I jump into the experimental data. What you can see in the nitrate data are this gradual accumulation of nitrate, likely due to nitrification of this ammonium that's coming from the wastewater treatment plant. On the plot of FV over FM, this is the quantum yield of photosynthesis, we see higher FV over FM up here, and then a real hotspot of low FV over FM in the lower river down here. These data were made with a phytoflash system in sampling surface water. But of course, there's cause for concern here. Phytoplankton are not doing as well down here as they are up here, and that's reflected in some of the chlorophyll data you saw from Tamara. So this setup, where we have no ammonium up here, relatively a lower concentration of nitrate, and higher nitrate and higher ammonium down here, is a perfect setup for the design of an experiment. And that's what we sought out to do. So we took surface water from 2 stations upstream of the wastewater treatment plant. Station A, which is upstream of the confluence with the American River. Station B, which is upstream of the wastewater treatment plant. This was not from one of the regular stations. We wanted to be sure we didn't get any sloshing up of ammonium from the wastewater treatment plant going upstream on a tide. So we were well above that. And we had our underway ammonium and underway nitrate, so we could tell that we had not yet hit the pulse of ammonium when we were sampling at Station B. And then Station C, located directly downstream of the wastewater treatment plant outfall. And so we took water from each of these stations and amended it with a series of treatments. And these treatments were placed into 10-liter cubitainers that were then incubated at 2 different light levels. So that's where this is going. So here are our treatments. Here are stations A, B, and C. And then for each of these stations, we had 4 treatments for A and B. An unamended treatment, a treatment to which we added clean ammonium, intended to mimic the concentration that was received in the downstream water from the effluent. A treatment that, to which we added clean nitrate, which was intended to raise the concentration to be equivalent to what we were seeing downstream, and effluent obtained from regional San. And part of the goal here was actually to test a hypothesis about whether there was any difference in growth on effluent and growth on clean ammonium. Is there something else coming out of the effluent that might be influencing the phytoplankton community and causing the low FV over FM or otherwise suppressing growth? So that was one of the goals of our study, to test that hypothesis. And we ran all of these 10-liter cubitainers in triplicate. Now, grazing is something that we have been talking about a little bit today, and there were no clams in these cubitainers, and there also were no large zooplankton. We had a 300-micron screen that all this water was filtered through. So we incubated all of these cubitainers at 2 different light levels. One light level, which is called the highlight treatment, was at 52% of incident irradiance just below the surface and held at that light concentration. The other, which we'll call low light, was at 6% of irradiance. So you heard a little bit about in the previous mesocosm study that we were incubating, the mesocosms were incubated at surface light conditions, and they saw a lot of growth. Here we're explicitly testing different irradiance levels and the effects of those different irradiance levels on phytoplankton growth and community composition, right? As well as the nutrient amendments. So what do these light levels mean? Well, we had light profiles at each of our stations, and here's what our high light and low light levels are equivalent to in a well-mixed 10-meter water column in the river. So the high light level is equivalent to being held at about between 0.65 and 0.8 meters depth. That's where you hit about 52%. The low light levels are equivalent, and they're different at each station because the PAR profiles were different at each station, to roughly 3 to 3.5 meters depth. So a better illustration of what phytoplankton might be seeing is this, right? This is what the water column looks like. So our high light level phytoplankton are being held here, and our low light phytoplankton are being held here. They're not being exposed to these light conditions, even though in situ, in the environment, they would be being mixed down into this system. So how well did we do? Well, we did pretty decently at mimicking the concentrations. Part of this is pretty hard. You have these underway systems, and you're measuring ammonium and nitrate, and then you have to amend all the experimental treatments to match those concentrations exactly as you can, right? So how well did we do, especially since you're handed effluent and told, here's roughly the concentration in the effluent, we think. So we did pretty well. This was downstream water, had about 57 micromolar ammonium, 2.2 phosphorus, and about 8 micromolar nitrate. At station A, when we added the clean ammonium, we got to 55, and when we added the effluent, we got to 45. So our effluent was a treatment that wasn't quite equivalent to the ammonium levels here, but pretty darn close. The effluent, it turns out, contains a little bit of phosphorus as well, and our nitrate, we were able to increase to roughly the target concentration. So what did we do? We got to hang out on a houseboat in the Rio Vista Marina for 48 hours and set up our incubators on the side here. And we measured every 12 hours chlorophyll A, POC accumulation, changes in nutrients, and then used the stable isotopic tracer method to look at ammonium and nitrate uptake rates. And then initial and final samples also had phytoplankton enumeration from microscopy. So this is our experimental design, and I'm going to walk through all of the results for you next. So that's what's happening. All right, chlorophyll accumulation. So to orient you here, station A and the different treatments are here. Station B and the different treatments are here. And station C already has the treatment in it, right? It has the effluent. It's downstream of the effluent pipe. So this is under high light conditions. After 48 hours, we see a lot of chlorophyll accumulation, whether we add effluent, whether we add ammonium, or whether we add nitrate. We're seeing growth of the phytoplankton communities. Much less so, we still see growth, but much less so at the low light level treatment. So what about carbon accumulation? Oh, sorry. Now, broken up over the 5 time steps, so 5 12-hour time steps, we see an accumulation of chlorophyll that's ramping up rapidly, right? So this was only a 48-hour experiment. We didn't run it out over 4 days. We didn't see the 40 micrograms per liter that some of the other experiments have seen, but we certainly see a trend across all of these treatments where chlorophyll is going up dramatically in pretty much every single treatment, including in this control that has no nutrients added to it, right? We see this growth here under the highlight treatment. Under low light, we see less growth, but also this upward trend. And we can calculate growth rates, et cetera, from all of those data. So in terms of carbon, this is net carbon accumulation. So this is the delta of the POC over time between 48 hours and the initial start. So we see carbon accumulation, less so if we don't add nutrients to this low nutrient water from site A, but we see carbon accumulation pretty much equal across the board in all of these highlight treatment samples. We see a lot less carbon accumulation if the light level is lower, equivalent to 3 to 4 meters down in the water column as opposed to 0.8 meters. Let me do that again. This is the difference from only light. So looking at cell density and chlorophyll per cell, we can get into all of these details if you want, but you see cells increase. Cell density increased in all of these treatments over the 48 hours at both light levels, but increased more at the high light level relative to the low light level. And if we look at chlorophyll per cell is generally higher in the low light than in the high light, which is nice to see. For nutrient drawdown, here are the ammonium concentrations in each of our treatments. There's obviously not much ammonium in the unamended upstream samples, right? If they don't have effluent or don't have ammonium, then they don't have much ammonium in them. But what we see is drawdown over time, as you'd expect. They're not using all of the ammonium over this short 48 hours, but they're certainly growing on it. They're certainly using it in each one of these treatments. They're using phosphate, but phosphorus is not becoming limited in any of these treatments, although they're certainly using quite a bit of it, and there's not that much of it to go around. And they're even using a little bit of silica, and we'll start talking about diatoms and community composition in a moment. All right, so uptake rates. This is part of the key point of doing this study, right? Do we see that the presence of ammonium inhibits or suppresses nitrate uptake rates, right? So here are our specific uptake rates for ammonium at all of our highlight samples at initial and final times. These are calculated through stable isotopic tracer methods, and we measure the unamended deltas as well. And we have lower uptake rates in the low light treatments, but again, we're seeing ammonium uptake in all of these samples. What about for nitrate? Well, it's exactly as you would expect. When ammonium is present in high concentrations, whether from the effluent in situ in the river, or whether we added it from effluent or ammonium, nitrate uptake rates are basically zero, but nitrate uptake rates are rather high when you don't have ammonium present. This we would expect biochemically, right? This is ammonium suppression of nitrate uptake at high and low lights. So we can calculate the rows, the actual uptake, by multiplying the specific uptake rate by the concentration of particulate nitrogen. And for example, at station A, when we add ammonium, we see basically no nitrate uptake, no nitrate uptake, no nitrate uptake, no nitrate uptake. All right, so what's going on in the phytoplankton community? I've just walked through the nutrients and the chlorophyll and the carbon and the uptake rate results, but what's happening in the phytoplankton community? I know the colors here are pretty tough to see, but to answer one of Amy's questions from before, the community is dominated by diatoms, and the next most abundant taxa are chlorophytes, and we do see some cyanobacterial species in these treatments. So this is station A, station B with all the different treatments. What you'll see here is that the phytoplankton count is going up dramatically from the initial across the board, and that the relative contribution of diatoms it actually increases a little bit over time and in a statistically significant way, but diatoms are certainly the dominant group in all of our treatments, and that remains true whether they're growing on nitrate, ammonium, or effluent. Here's the same results for low light, and actually under low light conditions we see slightly more increases in cyanobacteria at station A in terms of their relative abundance. This actually, it says phytoplankton community, this just breaks out the three most abundant species, Anabina, chlorella, and in our samples, Thalassiocyra. And takes a look at the relative cell counts of Thalassiocyra, chlorella, and Anabina as sort of species that are representative of these three different taxonomic groups. And again, here we have a strong dominance of diatoms throughout. Notably, in highlight, we don't have any Anabina in the initial or final downstream samples from the downstream river location, whereas we do in all of the others. And this is for low light as well, where we actually did see some Anabina and greater contribution of cyanos under these low light conditions. So in conclusion, and I don't want to speak to the, this doesn't solve the sort of question in charge of the workshop, because we, can different experimental conditions explain the vastly different results that we see? In this experimental condition, in one season, in May of 2015, over 48 hours, where we manipulated light conditions, all that was required to get the phytoplankton communities in the river, which were not at high concentrations, to grow, to fix carbon, to accumulate chlorophyll, all that was required was to raise the light level, and we saw dramatic differences between our high and our low light levels. Again, we can't speak to grazing impacts here because of the exclusion of grazers, and there's perhaps some evidence that at our upstream site, some of the reasons that we saw growth even without any amendment, even at our low light levels, might have been because we're excluding grazers. So we can't test that with the data that we have, and that's a full caveat. But do we see a strong effect of light on the phytoplankton community? Not its composition, but certainly its growth. So in summary, phytoplankton communities in the lower Sacramento River grew well on wastewater effluent, clean ammonium, and clean nitrate when applied with sufficient light to grow. Communities exposed to higher light levels grew more quickly than those to lower light levels, and diatoms are doing fine under these conditions, increasing the relative fraction of the community and certainly maintaining it. So what's next? The change in nutrient form that's coming, this 1/3 to 50% reduction and conversion to nitrate is unlikely based on these results to solve the ecological woes in pelagic organism decline. And the effects of what's going to happen are strongly related to light, right? So if we do continue to see the deepening of the photic depth in Suisun Bay under climate change, under flow regime changes, with the reduction in sediment loads from mining conditions over time, we need to think about the interaction of nutrient forms, nutrient abundance, and light, and it's really key to studying all these interactions together. So here are my acknowledgments, and thanks.
Thank you very much. Yes, if you want to applaud, that's great. We always appreciate that. Antoinetta, you're on the phone, so we're going to give you first crack this time. Oh, she's gone?
She had to go to an appointment. She'll be back later.
All right, great. All right, it's all you guys then.
I guess the question, the only question I have, I mean, seems pretty straightforward. The only question I had was in your last statement, or one of your last statements, where you said the interaction of light with the different forms of nitrogen. You want to elaborate on that?
Sure, I'm happy to.
Why do we need to be concerned about that?
Well, there are a couple possibilities, right? One of the things we need to be concerned about with light limitation being reduced over time is the question of how much does it have to go down? How much deepening of the photic depth will actually trigger the other potential fear, right? We have this concern about pelagic organism decline, but don't forget about the top half of Dave Sen's conceptual model where there's a fear of potential eutrophication at these high nutrient levels. So how much change in the light environment and the change in turbidity and availability of particular organic matter, or particulates, suspended particulate matter, is going to actually influence the ability of blooms to happen. We had a big bloom this spring. The other question is, will the amount of DIN that is continuing to come into the system be sufficient to support that production if light exists? I think I'm certainly convinced that the phytoplankton that are present in the system are capable of growing on the nutrients that are there, and that doesn't really matter, according to these results, whether it's ammonium, effluent, or nitrate. They can grow.
Yeah, that's fine. You had made a statement that we need to be concerned about light and the different forms of nitrogen interaction. I mean, we all know that the uptake of nitrate requires a lot more energy than ammonium. I think your results very much reflect what we see in other estuaries around the world.
They do. I think where that comes in is a question of nitrification, right? We have a bacterial community that, and this is one of those, going back to the points of agreement and disagreement, I think there's wide agreement that the lower Sacramento River has really active nitrification. You see this gradual increase in nitrate over time, or over distance coming down from the wastewater treatment plant. If all of that is coming out as nitrate, then this interaction with the benthos is fundamentally going to change if you don't have ammonium there present. Does that influence anything? I don't know. Gets back to the point about needing to incorporate the bacterial community into some of the future modeling efforts as well. So the forms certainly do matter to some of these microbial processes.
Speaking about another form, the cyanobacteria were a relatively small component based on...
Correct.
And I was interested in the last set of slides where you were showing specifically Anabina.
Yeah.
And speaking of different forms, do you have any sense of whether these are actively nitrogen fixing or not? And if so, how do you view the role of nitrogen fixation and when you're talking about different forms of nitrogen and that role in the system?
Yeah, so certainly if you're getting these higher concentrations from what I know about nitrogen fixing bacteria and heterocyst formation and all of that, if you have high concentrations of ambient DIN that's available, you're not necessarily going to get high rolls of inputs from nitrogen fixers. If we were in a situation where we were going to shut off all flows of DIN into the system entirely, then nitrogen fixation, I think, becomes an interesting question. But that, my understanding from what's the changes that are going to take place from regional SAN is you're going to get less overall loading, but it's going to be in a nitrate form. I'm not familiar enough with Anabina and the cyanos to speak to whether that could come through here. I will say there's a lot of question about sampling technique for chlorophyll and how we grab these, and so we were pulling. Our cubitainers were filled from just under surface water, so not 1 meter down, and I think it's really important as we move forward to pay very close attention to these sampling designs when we're looking at community composition and chlorophyll. Surface and bottom have an incredibly different light environment, incredibly different interactions with the benthos, and certainly with some of these surface cyanobacteria and some of the concern about those organisms.
Yeah, just a comment on the cyanobacteria. You just counted the big guys, not the picoplankton?
Right.
Okay. Because it could be that the picoplankton actually are important, but they didn't get counted like they do in most monitoring programs.
No, so we had a flow cam set up, but I wish that we had better coverage of the picos and had done all that from this, but we didn't have it. Yeah.
So you just quick follow up on the nitrogen fixation and the high concentrations of nitrate and ammonium, which likely are inhibiting that. But if DIN in the form of nitrate and ammonium were to be drawn down substantially to, say, non-detectable levels, then I would assume the Anabina are there as potential nitrogen fixers.
Absolutely, and looking farther upstream where we have relatively low concentrations, it's not below detection for either ammonium or nitrate, but they're low. They may be playing a role up there as well.
So from your experiments, it looks like the effluent doesn't have any contaminants that are severe. Is that right?
That's what we were looking at, and I didn't show, we measured with an FRRF, we measured FV over FM, and it was about 0.5 to 0.55 for all of these samples, regardless of the effluent or not, over time in our incubations. And so, we went in with the hypothesis that maybe we'd see this tremendous difference between effluent and ammonium, and it would explain some of the low FV over FM in situ, but it doesn't.
Right. The other question from our background reading is that if you have nitrate and ammonia together, then somehow or other there'll be more uptake and faster growth, faster than mu max, but that doesn't seem, you actually didn't do that. You had the clean ammonia, had the clean nitrate, but you didn't have the clean nitrate and ammonia, although you did have the effluent.
You hit on something that we certainly wish we had done, but we ended up, ironically of all things, being limited by these 54 10-liter cubitainers and the logistical limitations of actually running all of the uptake experiments from that. So I remember our original design called for 108, and it just couldn't be done. So we thought through that. We wanted to do it, but we didn't.
So what do you think? Do you think if you have nitrate and ammonia, then the growth rate isn't any different than one or the other?
Well, we do have one indication of that, which is our water from downstream of the effluent pipe. So that's not high nitrate, it's a little bit of nitrate. And those communities and their growth rates were not significantly different at all from any of the other treatments.
Right, which is what I expect. Last quick question. You took out the big grazers, 300 micron screen. There's a lot of grazers between there and 300. How important are they?
We have no way to directly speak to that other than we certainly saw them. when we were looking through the samples. So there certainly, it was not as if we had a complete absence of any grazers in these samples. There were grazers that made it through our screen and they were there. We didn't do any formal, that's just a fact of our sampling design, and we have no sort of formal analysis of the role that they played.
Right. Is it plausible in the future to, you did light, which is great, and showed it clearly, but probably there's a light-clam interaction here. So can you set up sort of reasonable, valid mesocosms to add clams in there too?
That sounds like a really great thing to come out of this workshop.
Anything else? Okay, thanks very much. We'll have the next speaker.
Next, we have Minée Berg from Applied Marine Sciences. She's done a lot of work, both field and lab, in the Delta looking at phytoplankton.
Yeah.
Minée.
Thank you. So I'm just testing, is this, oh, okay, that's the pointer.
You're going to have to hold the microphone closer to your face. There we go.
Can you hear me now? Okay. Thanks for inviting me to present the results of our study. The name of my talk is actually the name of a study that Rafe Cadella and I got funded to look into the impacts of ammonium on phytoplankton growth in Suisun Bay and the Sacramento River. And we had several collaborators that helped us with our study, which is divided into two parts. We had a field component and a culture component where we looked specifically at phytoplankton physiology. And we had also a person who helped us count phytoplankton from this area. And we were supported by a number of funding agencies, which I have a list of here, including the San Francisco Regional Water Board and the U.S. Bureau of Reclamation. So I'm going to be presenting the results from both the field component and the culture component today, and there's a lot of data, so I will probably be flipping through it pretty quickly to get through it, but I'm guessing these talks will be available to the panel, so you can look at it later. We are here to examine the ammonium paradox, and I just have copied the definition for the ammonium paradox as it was written in the paper distributed by Janice, the charge to this workshop. And basically, the paradox states that elevated concentrations of ammonium suppress nitrate uptake, and that this results in a lack of access to nitrate and lower productivity and growth. So this was developed in papers by Dugdale, Parker, and Glibert. And the reason that we're calling this a paradox is because it's well described in the literature that ammonium is preferred by phytoplankton. And as mentioned earlier, it's in reduced form. It's the form that you need to, the form that the cells can use directly for assimilating into amino acids and the synthesis of amino acids. So I have, as a motivation for our study, we wanted to test some of the claims that were made in these papers, basically how ammonium inhibits nitrate uptake. It's very well known and very well described in the literature, but in these papers they go on to say that it impacts chlorophyll accumulation.
I think your microphone's off.
Yeah, I think yes, it is. Okay, and then in Parker et al., so in Dugdale, they basically focused on how reduced rates of nitrate uptake is kind of synonymous with reduced growth rate because phytoplankton grow on ammonium very slowly. So in order to grow fast, they have to grow on nitrate. And then in Parker et al., it was a slightly different version of that, where they found that ammonium uptake declined as ammonium concentrations increased. So then the way that was interpreted basically is that reduced ammonium uptake leads to reduced rates of primary production and therefore growth. And these concepts were put into a theoretical framework by Glibert et al. And this paper was published in 2011. And here, this theoretical framework was used to describe ecosystem interactions in an idealized estuary with riverine influence, and where there was a large point source, a discharge of ammonium to the river, and how this point source discharge is transformed in the river and transported downstream into the estuary, and how this impacts the community, including plankton dynamics. So basically the way that this theoretical model works is that there's an immediate impact zone where there might be direct toxicity effects from ammonium and indirect inhibition effects, and these inhibition effects would be what is described by Dugdale and Parker, basically reduced uptake rates and productivity and growth. And then after the immediate impact zone, which is up here, there is this river mixing zone, and here you have, whereas up here there's organismal avoidance, down here you have the organisms appearing. And you might have, if you read the legend to this figure, you might have some cyanobacteria that grow in this portion, and then you move down to the estuarine filter. And this is a place where the ammonium is transformed to nitrate by nitrification. You have increased availability of nitrate. And here you might have diatoms occurring and even blooming. And the reason, as described in the paper, why you might not have diatoms anywhere else is that diatoms physiologically prefer, and in some cases require, nitrate over ammonium. So we thought this kind of theoretical framework would be a great platform for collecting data and trying to validate this model. And so we decided we wanted to, we're substituting the Sacramento River and the Sacramento wastewater plant discharge as this high impact zone here, and then we wanted to look at the processes going downstream and into Suisun Bay. So the goals of our project were, the first goal that we had was to see if we could measure the impact of ammonium on phytoplankton physiology directly. And when I use the word directly, what I mean is that when there's high or low concentrations of ammonium right around the cell, it has an impact instantaneously on the physiology. So that impact will then be manifested in chlorophyll after some time. So if you're in a system, let's say Chesapeake Bay, where you have chlorophyll of 10 microgram per liter, and the phytoplankton are growing rapidly, they're dividing once a day, the impact of changes in nutrient concentrations could be seen already after a day. you would have chlorophyll going from 10 to 20. And you can measure that. In Sacramento River, where, again, to compare it to Chesapeake Bay, where you have light attenuation coefficients per meter that range from 1.5 to 4, you have division time of 3-4 days. So it takes like up to 4 days for the cells to divide. So also starting chlorophyll concentrations are between 0.5 and 1 microgram per liter. So over 4 days, you're going to see an increase from 0.5 microgram to 1 microgram of chlorophyll per liter. And that is at the limit of detection for the chlorophyll analysis method. So it's really hard. So FE or FM is a method that you can use to measure impacts on the physiology of phytoplankton at the same time scale as they're occurring, basically instantaneously. So we wanted to look at whether we could show that there was a dose-response relationship between Fe or FM and ammonium concentration. So that was our first goal. And secondly, we wanted to examine whether diatoms from Suisun Bay and the Sacramento River grow faster on nitrate than they do on ammonium, and the corollary to that is whether non-diatoms grow faster on ammonium than they do on nitrate. And we wanted to examine the concentration of ammonium needed to inhibit growth in phytoplankton from the region, and to look at the relative roles of changes in nitrogen source versus changes in irradiance. And also, lastly, this is basically our field component to investigate how the abundance of diatoms and other phytoplankton are related to environmental variables going from the immediate impact zone in the Sacramento River downstream and into Suisun Bay. So Fe or FM, another way to describe that, what it actually measures is the quantum efficiency of photosystem 2. So basically, a phytoplankton cell, or any photosynthetic cell, they convert solar energy to chemical energy. And this chemical energy is used to drive carbon fixation. But if there is changes in carbon fixation, the cell has to really adjust its conversion of solar to chemical energy really fast, because it releases electrons into the cell that can fry the cell, literally fry it. And so there is a very close feedback between carbon fixation and this process here of converting solar energy to chemical energy. And so anything that impacts carbon fixation is going to impact this process here. But you can also have direct impacts on this conversion by competitive binding to the photosynthetic apparatus of toxins or whatnot. Okay, so that was a little bit of background on why we want to use Fe or FM. So here are the methods that we used to answer our first question. We isolated phytoplankton from Suisun Bay and Sacramento River. And we used single cell isolations. And the reason that we went to this trouble is that we wanted to investigate these impacts under controlled conditions so we could be sure that we were measuring changes to phytoplankton physiology and not ecosystem effects. And we didn't want to use cultures from culture collections that may have been in culture for a very long time and change their responses. And also we wanted to use phytoplankton that were endemic to the Delta region and not from, the East Coast or somewhere else in the world. And then when we got these into culture, we maintained them on filtered river and bay water at conditions that were very similar to what the conditions when we isolated them, so at the time of isolation, in terms of irradiance and salinity and temperature and nutrient concentrations. So we maintain them in culture at ammonium concentrations between 0 and about 8 micromoles per liter, and nitrate around 60. And then we measure, as soon as we had them into culture, we started our experiments, and we tested ammonium concentrations from 20 to 3,000 micromole per liter, and then we measured the FeORFM, which is the variable over the maximal fluorescence, and we also calculated percent suppression in FeORFM in response to the changes in ammonium. So here is a list of the cultures that we isolated and used for these experiments. And before we started the experiments, we did some preliminary investigations to see how they responded to ammonium. And in those preliminary investigations, we figured out that there were really only 2 species that were sensitive to ammonium. And that was the 2 top species. These are diatoms that we isolated from the Sacramento River. And then we also isolated 2 diatoms from Suisun Bay, and they were not very sensitive to ammonium. And these diatoms were Entomones paludosa and Thalassiosyra weissflogii, and these are both dominant in Suisun Bay. And I'm going to show phytoplankton community composition data just a little bit later. And then we isolated 2 non-diatoms, chlorophytes, chlorella, which is very dominant in this system, and it's about 2 microns in size, and radiococcus. Okay, so here are the results of the ammonium dosing experiments. So what you have on the y-axis is the percent suppression in the FeORFM, and on the x-axis we have the day of the culture. And you can see that already on day 1 there is significant suppression in the FeORFM, and that was in both of these cultures, and with maximal suppression around day 2, depending on the nitrogen concentration, or day 2 to 3. And then another way of looking at this is looking at the percent suppression as a function of the ammonium concentration. So here you can see, so here the different colored dots are day. So you see that on day 0, there's not much suppression going on, regardless of the concentration, but it really ramps up between day 0 and day 1. And then in Fragillaria and in Astronella, it kind of ramps up every day a little bit more. But you notice here, at these lower concentrations of nitrogen, there is really no suppression. So what we concluded from these short little experiments was that changes in FeORFM really provide a reliable method of detecting ammonium toxicity or inhibition response at the same time scale as it's occurring, and that there was a suppression in FeORFM in a dose-dependent manner. Okay, so now moving on to the next questions that we wanted to answer. Do diatoms grow faster on nitrate than on ammonium? And at what concentration of ammonium is growth inhibited? So here's just a short method slide. Basically, we grew all 6 different species on diatoms on 5 different concentrations of nitrogen at a constant irradiance. So this irradiance here, well, it says micro-Einsteins there, but it's 85 micromole photon per meter squared per second is pretty typical, maybe a little bit high of average light intensities in the water column. Okay, and we also wanted to do a test of low addition of nitrogen, and we actually added 5 micromole either as ammonium or nitrate. But it's hard to see a difference in cell abundance in terms of calculating growth rates from such a low addition, so we measured primary productivity. And I'm not presenting that data here. Okay, so here is, okay, so going back to this graph. So we had 6 different species, we had 5 different nitrogen concentrations, 2 sources, we have basically 60 different cultures. So I'm not going to show 60 graphs here. This is just 2 graphs to kind of give you an idea of the typical time course of the culture growth. And these graphs here represent chlorella cultures at low and high additions of ammonium. And I actually have relative units here on the y-axis and then the day on the x-axis. And you can see chlorophyll and cell abundance go up, ammonium goes down, Fe or Fm is pretty constant. You see a maximal value in this black line here represents mid-exponential growth. And so at this point, we kind of, this is the Fe or Fm value that we used for our analyses, and we also collected samples for measurement of carbon fixation on that day. So here are some of the Fe or Fm results for all the cells, for all the cultures. So I have the chlorophytes here in panels A and B, the estuarine diatoms in C and D, and then the freshwater diatoms in panels E and F. And then the dark bars are ammonium, the light gray bars are cultures grown on nitrate. And then the x-axis is nitrogen concentration at the start of the time course. So you basically see very little differences in the Fe or Fm with growth on different nitrogen sources, very subtle differences, but the main difference is for the freshwater diatoms, obviously, where you see the decrease when they were growing at higher concentrations when they were growing on ammonium. So here I have growth rates in the same cultures, and these are calculated based on changes in cell abundance. And here you actually see significant differences in the growth rate with nitrogen source. So this chlorophyte here, chlorella, grew better or faster on ammonium. This chlorophyte here grew faster on nitrate. The estuarine diatoms grew faster on ammonium. And the freshwater diatoms grew faster on ammonium at very low concentrations, and then obviously at higher concentrations, growth was inhibited. All right, so we wanted to know how do our results compare with other culture studies. And so I went into the literature and I researched differences in growth rate on ammonium and nitrate in a whole host of phytoplankton cultures, and there's actually quite a bit of data out there. So here on the white axis, I have the growth rate difference. It's basically the growth rate on ammonium over nitrate, and the difference expressed as a percent. So everything that's above this line here means that growth rate of ammonium is faster than a nitrate, and everything below growth rates of nitrate are faster than on ammonium. And this little dashed line here is, I think it's the mean difference, which is around 14%. So one thing that struck me looking at this graph is that these differences are really species dependent. You know, it's not like you can say, oh, all the diatoms, you know, fall here, all the chlorophytes are over here. It really differs depending on the species and also on strains. So that was pretty interesting, and that's exactly what we saw in our study too. You really can't make any generalizations based on taxonomic divisions, I think. Okay, so here we concluded that, you know, in terms of answering our questions, do diatoms grow faster on nitrate than ammonium? No, it was really species dependent. But in this study, where we isolated the diatoms from Suisun Bay and the Sacramento River, we found that actually all of them grew faster on ammonium than nitrate. We also, you know, comparing our diatoms to the chlorophytes, we didn't find that the one chlorophyte that grew faster on ammonium than nitrate, that the difference was greater for the chlorophyte than the diatoms. So that was interesting too. And we found that in the 2 species where growth was inhibited by ammonium, it occurred at concentrations. So if we take inhibition to mean a reduction in growth rate by 50%, we found that this occurred between 300 and 700 micromolar ammonium per liter, which is quite a bit above the concentrations that you normally come across in the Sacramento River and Sassoon Bay. And then the next thing we did was to look at the interaction.
You have just a couple of minutes left, okay?
Oh, I do? All right,
Yeah.
so we also looked at interactions of light and nutrients. And I'm going to skip pretty fast over that. So I think that the field data is also pretty interesting. So what we did 4 cruises in 4 different seasons. And we started out in Sassoon Bay, and we went up from Sassoon into the Sacramento River, and we sampled 32 stations, and we measured a whole host of variables. And so I have a list of the variables here. And we hit some of these key stations that there's a lot of historical data from them. And so I'm only going to show a couple of the parameters that we actually collected. So this is just a little snapshot. So basically, if you look at the salinity, it's the same in the Sacramento River. So these stations on the right-hand side here are from the Sacramento River. And these stations, starting with station 19, which is Rio Vista, are from Sassoon Bay. So you see that there is a pretty big difference in salinity according to season in Sassoon Bay. Here's the nitrate concentrations. You see this ramp up of concentration with higher concentrations in Sassoon Bay compared to the river. And also quite a bit of seasonal difference. Chlorophyll A, you see this linear decrease going down the river starting from upstream. This is actually quite a bit upstream of the wastewater discharge. And ammonium, you see a ramping up in concentrations right at station 27, which is where the Sacramento wastewater effluent is discharged, and then this decrease. So here are results of changes in FE or FM with season. Basically, I think the takeaway here is that FE or FM really changed a lot based on season. In August, it was the lowest. FE or FM was only 0.2, and that is really, really low. And then it was highest in January, where the median was above 0.4, but also at certain stations in October where you hit FE or FM of 0.6 and above 0.6. So really very, very variable. And here is a summary of the FE or FM across all 4 seasons. We found, which we thought was pretty interesting, that you have these high spots and low spots. So above, this is the I-80 bridge. So here you have pretty high FE or FM. So most of the FE or FM was between 0.4 and 0.2. And then you have these low spots in the river. It's here at this station 25, which is Hood, and here at station 20, which is right above Rio Vista. And then the high spots in Sassoon Bay were Honkers Bay, station 15, and this is DW11, Grizzly Bay. Okay, so here's the results of phytoplankton composition. So the diatoms, this is expressed as a percent phytoplankton composition based on biovolume. So what you can see at a glance is that the percent diatom composition is very high. Basically, the diatoms dominated from all the way upstream in the river to Sassoon Bay at all 4 seasons. There's really no difference. But the next graph here shows how the diatoms parse out. Actually, this graph shows all the different groups, including cryptophytes and chlorophytes and the various diatoms. But the take-home message here is that these the 4 stations to the right, where you have the red bars, those are in the Sacramento River, and the stations to the left, where you have the teal blue bars, those are all from Sassoon Bay. And then you have, these two stations in the middle, that's like the transition zone between the Sacramento River and Sassoon Bay. So this is station 19 and 17. So this is where like the transition zone ends. So this coral or this red color is Synedra. It's a pennate diatom called Synedra ulna. And then here you have Thalassocyra. So this is Thalassocyra dominated at all seasons in Sassoon Bay. So that is basically our conclusion, that the system is very much dominated by diatoms. And so we didn't find this model here proposed, or this theoretical framework proposed in Glibbert et al. To really jive with the data that we collected. So basically, we didn't see any toxicity or growth inhibition in the immediate impact zone where the discharge occurs upstream in the Sacramento River. And we also didn't see any changes. I mean, there were huge changes in chlorophyll, nutrient concentrations, and a whole bunch of variables. But the composition was pretty consistent between seasons. So that was a real surprise to us. So the diatoms, they basically dominated irrespective of ammonium and nitrate concentrations and ratios. And we did not see that the diatoms that we studied here require nitrate. So I don't have time to go over this, but
Yeah, we need to move on to the questions before lunch.
Yeah, so I will leave this. This will be available for the panel to read.
Great. So let's just take the next 20-minute period before lunch to give you guys the opportunity to ask questions of any of the presenters, anything you didn't get to in the morning earlier, or anything that sort of came up during the later talks. So feel free to call on any of the presenters as you wish. Yeah, and if the presenters can come up and sit at the microphones here at the table, just turn them on, and then everyone can hear you when you answer. Thanks. Yeah, right here. Just turn your mics on. All right, it's all yours.
Is Antionetta on the line?
I don't.
Not on the line, okay.
She told me she was going to be gone for a few hours.
Oh, okay. Okay.
Go ahead, Amy.
Okay. Mine, if I could ask a question, follow up. I do have it on. Okay. You made the statement that there may be differences, correct me if I heard you wrong, that there may be differences in growth among algae, among phytoplankton, taxa, on nitrate versus ammonium, but it's not necessarily grouped as diatom versus non-diatom. Did I understand you correctly on that? Okay. In the some old literature, and I'm thinking, there are other papers I'm sure, but I'm thinking one by Brian Moss in the early 70s where he looked at changes in growth rate among freshwater desmids, but he also included a euglenophyte. And one of his conclusions was there wasn't much difference in nitrate or ammonium, but but what there was there, he was speculating or was proposing that it was really grouped oligotrophic versus eutrophic, and that it made more sense that algae, regardless of what phytoplankton, regardless of whether they were diatoms or non-diatoms, if they were growing in nutrient-enriched conditions, they would be, in quotes, adapted to growing better on nitrate compared to, or ammonium compared to nitrate. Do you have any sense that among your taxa that there can be distinctions in terms of describing them as, in quotes, eutrophic versus oligotrophic forms? Or do you think that has anything to do with any of the differences you are seeing, not necessarily within diatom groups versus non-diatom taxa, but across those groups?
I think that in order to answer that question a little bit better, it would have been cool if I looked at differences.
Please speak into the mic a little bit because we're trying to record the session as well. Thank you.
It would have been cool to look but Pseudococcus, for example, is this on?
Should show a green light when you push
it
that.
Does show a green light.
Okay.
Now it does. Oh, now it does. Okay, sorry. Which we didn't do, We only isolated our species from Sassoon Bay. and the Sacramento River. And so we didn't look at species that would be representatives of those kinds of conditions.
Okay, so are the species the taxa you did isolate, were they dominant or not?
Yeah, so Thalassiosira dominates Suisun Bay, the Thalassiosira genus. There's several different species in the genus, but it dominates at basically all 4 seasons. This year that we did our study.
What about the Asterianella and the Fragillaria, are they?
So we saw Fragillaria in the Sacramento River.
Would you consider it rare or medium, high dominance?
It was like 6-7% when we isolated it, that cruise that we isolated it from. And so I see it, around that percentage. Yeah.
Of your list of 6 questions up there, can you pick one you think is really important?
Well, that would be, I think, number 1. So I think that basically differences in experimental conditions don't really explain why some people think that there is an ammonium paradox and others do not. And it's because people or the scientists are interpreting the results differently. I can look at results by Alex Parker, which I have in the 2 slides following this, and I would take that data and interpret it completely differently, the same data.
Right.
So I think really it boils down to the interpretation.
Okay.
Yeah.
Did you do the actual uptake, N15 uptakes, and compare them to growth rates?
You mean in the cultures or in the field?
Either one.
No, we didn't. But what we did do when we did the light nutrient culture experiments, we collected particulate organic matter that we analyzed for the natural abundance of carbon and nitrogen isotopes. So actually right now I'm looking, trying to interpret that data. And we are trying to calculate fractionation factors and look at how the natural abundance of isotopes change with different nitrogen forms and irradiance levels. And how that relates to growth.
Right. Sort of back to your methodology question. So the growth rates, your data and the literature show that typically their growth rate on ammonia is either equal or somewhat faster than nitrate for a lot of species, especially for here in the bay. So how could you get uptake of nitrate to be faster than ammonia if, in fact, for your species, you actually, in terms of growth. So we're looking at uptake versus growth, which is a methodology sort of issue, long-term integration versus this short-term isotope, dilution and regeneration and all those things.
So the question is, how does growth compare to uptake?
Right, and how could you, if growth is faster on or equal to on ammonia, how can you then turn around and do a short-term, N15 uptake of nitrate and show that it's actually faster than ammonia, and therefore the cells have to take up ammonia or take up nitrate?
Yeah. So basically, if I could paint a progression of control in the cell, I would start with light, and the light really controls the rate of primary production, which controls growth in the cell. And then that growth rate then dictates the amount of total nitrogen or the nitrogen uptake rate into the cell. So the cell takes up nitrogen from all the available sources. And so in the field, what you typically have is you have a little bit of ammonium there, a lot of nitrate, some urea. And so all of those 3 sources contribute to the total nitrogen uptake into the cell. And when there's no nitrate available, the cell cannot take it up. So when the nitrate concentration is drawn down in the field, you just don't have nitrate uptake. Right. So the uptake of the various nitrogen compounds depend also on their availability in the water.
Right.
So you have to interpret the nitrogen uptake rate in relation to what's there to be taken up. So you can have way higher rates of nitrogen uptake if there's no ammonium. You can have way higher rates of ammonium uptake if there's no nitrate and ammonium is available, rather. Yeah.
Thank you.
I wanted to compliment you and others really on the work that's been done. I think it's really very good and definitive. The inhibition that you noticed in the freshwater diatoms, is that you think that's a pH effect because of the medium not being buffered and that the pH got higher and therefore there was just a lot more unionized ammonia at those high concentrations?
It could be, but I think that I don't think that it was such a big effect because we measured the FeOFM in all these cultures. And so, when we were growing them in this medium, we basically had a FeOFM of 0.6 and above in all these cultures. So if there was a pH effect that affected their rate of growth or primary production, we would have seen it in the FeOFM, and we didn't.
Well, pH effects, though, can be quite, the effect of ammonia toxicity, I don't know that much about it, but it's a pretty broad effect, isn't it?
So I actually read a paper referenced, I think, in a review paper by Paul Harrison. Yeah, that. So I had a real hard time trying to find papers on the toxicity effect, like exactly how does it manifest. So this paper written by Droth on cyanobacteria argues that ammonium toxicity is a direct effect on the oxygen evolution complex in PS2, and that it competitively binds with oxygen. And yeah, so it's pretty direct.
Well,
in phytoplankton.
It's probably pretty irrelevant anyway because those concentrations are really very high, I think.
Yeah, if you think about it, in order to get the FeOFM down to 0.2 as it was in Suisun Bay in August, which is dominated by Thalassocyra, you would have to have like an ammonium concentration of 3,000 micromole per liter.
So why is the Fe or FM way up in the river at the very top so high?
Well, I have a graph that could potentially explain that. Two slides after this. But basically, the Sacramento River is, there it is. Thanks. The Sacramento River is dredged. And so it's dredged up until river mile 56, which is a little ways above where the Sacramento Regional Wastewater effluent is discharged. It's a few miles above that. So if you look at that graph, on the top graph there is what I call the depth deviation. So I've taken the average depth of the Sacramento River all the way from down by Rio Vista to up where the I-80 bridge crosses. And then anything that's the numbers above means that the depth is less than the average, and the numbers below the line is a depth that is much deeper or deeper than the average. So you see this changeover where you go from 3 to -3, so there's a 6-meter change in the depth. And then the light attenuation coefficient is around 1.5 to 4, so your euphotic zone depth is like 1 meter to 2 meters. And so what you have is the lower graph is the euphotic zone depth as a ratio of total depth expressed as a %. So what you have is this huge change from the phytoplankton being 80% of their time in the euphotic zone upstream of river mile 56 to 20% or less below river mile 56 in the euphotic zone. And so those black dots there are primary activity measurements made by Alex Parker. So you see how they vary with the time that phytoplankton spend in the euphotic zone.
Right, so that's a great example of the importance of light.
And the importance of how you interpret your data.
Right.
Let me put a general question out to everyone who spoke this morning. I think we can safely conclude that there's a nitrogen issue, right? Is there general agreement that nitrogen, well, that there's a nitrogen issue? And if so, what are the issues that you can identify to us at this point? Setting aside the ammonium inhibition thing, which you've addressed, but I mean, clearly there is stimulation by nitrogen relative to phosphorus in the system. Why should we care at this point? This is just a general question that I'd like to just put out there to everyone.
So, sorry, what do you mean by stimulation of nitrogen relative to phosphorus?
Well, I mean that the bioassays and also the evidence that we've been presented shows that there's a nitrogen effect most of the time in terms of stimulation or eutrophication potential anyway. Dave, you look like you're ready to jump on that one.
I think there's probably a number of ways that, number of concerns that could be raised. One of them that always comes to mind for me is the idea that if we could think about the nitrogen abundance or nutrient abundance in general as like kinetic energy versus potential energy, where the massive amounts of nitrogen in the system are potential energy that are they're restricted from being turned effectively into phytoplankton biomass by combinations of physics and biology. And we know that the physics and biology are changing. And so and constantly changing. And so the biggest issue, one of the biggest issues I think, is the anticipated future issues that could crop up. And in a much more, and I know that's a big, broad question, and it's one we're also wrestling with in the Bay. More specifically, and I think it's something that we've seen recently, is with increasing frequency or apparent increasing frequency of microcystis blooms, that as the nutrients are there, they may not cause the bloom to occur. It might be the lifting of the biological and physical factors. But once those are lifted, then organisms are able to tap into that large nutrient reserve and take off to larger levels. So those would be two concerns. The third, I could imagine, is and this is something I know very little about, but the macrophytes and the extent to which uptake by macrophytes or the availability of nutrients is influencing the abundance and spatial coverage and abundance of macrophytes.
So is this on? Yeah. So I'm trying to understand your question. What is the issue with nutrients? I mean, again, I think the paradox really here is that in most systems, high nutrient levels lead to eutrophication. But in this system, we have high nutrient levels, but we don't have eutrophication. We have concerns that we have too little phytoplankton productivity. I think going forward, we are going to see a step change in nitrogen inputs to the delta by 2021. And so really the question is, if we lower the nutrient concentrations, are we going to enter a new realm where the concentrations of nutrients are going to affect phytoplankton in a way that they aren't affecting it now? And potentially, I would say there's some potential for some regions of the delta to become nutrient limited. Again, there's a lot of questions to see how nutrients are interacting with aquatic vegetation. And when we mean aquatic vegetation, I think a lot of times we're thinking about the invasive aquatic vegetation that people are trying to prevent. But of course, there's the good aquatic vegetation that's going to be built into the wetland restoration that will also draw down nutrient concentrations, but also could be ways that nutrients can be recycled in ways that we don't understand right now. because they can, they're obviously plants grow and die, and they also can provide areas where maybe you have more nitrogen fixation by some of these communities that can grow in shallower waters. So I think that looking forward, the real question is what the step change and decrease in nutrient concentrations are going to, how that's going to impact regions of the delta, and how that relates to the hydrology, really, because we have lots of different habitats with lots of different water residence times and light fields and all those other drivers that show up in people's conceptual models.
So is it fair to say that there really isn't a nutrient problem, but the problem is actually in the flow of energy or biomass now into clams rather than flowing on up into the higher trophic levels? And so by taking nutrients down to a new lower level, you're not going to actually be able to stimulate these PODs, these pelagic organisms. So it's still going to be less food to move into the clams. Is that right?
Well, again, that's that's why I think SFEI's loads and trying to figure out what, I don't know if we know that we're going to be nutrient limited enough to cause a decline in primary production. I think that's definitely a question that's out there, and I will leave any interpretation of whether clams are gonna play a big role to Jan's talk later today. Let her speak about that, but there's definitely, that's still a big factor on the table, is what the role that clam grazing has, as well as I think the changes to the hydrology that the system's experienced over the last couple of decades.
So this is sort of summarizing what was just said, but on the one hand, there's the specter of eutrophication and harmful algal blooms. And on the other hand is a question about lower nutrient loads leading to nutrient limitation. And we need effective coupled models and experimental monitoring that can allow us to sort of distinguish between those potentials. I think the nutrient loads, from what I understand, are not going to be so low that given our current light levels, we're going to see nutrient limitation soon, but it's certainly something to think about. I think there's a lot of work to be done because of the specters of harmful algal blooms and eutrophication and what they would mean for the system that hasn't had those to this degree. Certainly there are microcystis alarm bells going off around the country and around the world right now, and you can hear some of them here.
So I have a little prediction, and that is that in order, well, the way I see the system is that this is a system that has high turbulence, and it's very conducive to diatom growth because you have high turbulence and high nutrient concentrations. So and cutting the nutrient discharge by 30%, given the very low levels of biomass in the system, whether that's going to bring it to limitation, I doubt it, but if for some reason the turbulence is reduced, either due to climatological changes or reduction in flows from the building of a tunnel, something that causes the water column to stratify, and then you get a top layer where you don't have light limitation and you get complete drawdown of nutrients, I think that's when the community would potentially change radically away from this diatom-dominated system that we have, and you would see potentially really increased growth of cyanobacteria all through the system. So I think the turbulence right now is like the saving grace.
Just to add on to that, I think we finally heard the S word, which is stratification. And we've been sort of talking about this so far as if there's just this ongoing secular lightening of, deepening of the photic zone that we should anticipate into the future. But we haven't talked about the changes in flow regimes associated with water usage and climate change that might do exactly what Monet just said. And that's something we really need to get at.
We probably have time for one more question, if you guys have any.
do we know what the predators are on clams? In other words, do we, besides controlling nutrients, do we need to somehow or other look at control of clams because that flow of biomass is going into them rather than higher trophic levels?
I wonder if Jan Thompson would be able to answer that. I have no idea what controls
Dan or
clams.
John, anybody? Oh yeah, ducks.
But it seems like they're not doing a very good job.
It depends where you look. They do a great job in San Pablo Bay.
But in the shallow areas, or?
Yeah.
Yeah, okay.
Okay, thanks very much. So we've had a good morning session. We're now going to take a lunch break until 1:20. We'll start promptly at 1:20. And Janice, do you have any instructions or exhortations?
No, I just wanted to make an announcement. I hope you come back after lunch, but if people are not, if they're slipping next door or something, just want to let you know, we are gathering back here tomorrow. The panel is going to be working very diligently for most of tomorrow, but we agreed to have a check back in session. Please don't expect lots of conclusions at that point, but maybe some early thoughts, and I'll have a better idea then. We'll talk about what the next steps are after this white paper's done. But I am moving the time up. One of our panel members is actually leaving tomorrow evening. So to enable him to get to his flight, be back here at 2:45 instead of 3:15. That's tomorrow afternoon. And we'll end by 4 on tomorrow. Thank you.
Okay, thanks.
Yeah. You guys did a great job.
I cleared out the hardest room. I was ready to go. I was like, oh my God. You helped him towards the mirror and you haven't played for a while. She's like, oh, I forgot I was actually playing with her.
Congratulations.
I know you have lots more.
Thank you very much. I went wrong.
Yeah.
Thank you. I am.
Such a problem with the together.
I did a good job, by the way.
Thanks.
Yeah. Good job.
Thanks. Look forward to talking with you more about all this.
So I remember writing a proposal in the Senate 15 years ago. Title was, "Is nitrogen nitrogen?"
Oh, yeah?
Well, the history in North Carolina, so we see this about
Science, does it translate into lower production, or is it just they start growing on the ammonium and then they switch?
Well, they just use ammonia whenever they can. But we have a lot of coupling of regeneration.
One of the things, in response to your question about not that this is going to happen anytime soon, but if, well, let's say both nitrogen and phosphorus loads decrease dramatically and in proportion. In terms of what is most effectively stored in the cell, it's something you can do. And so then potentially create a situation, going back to the other panelist question about,
Fixation.
no, not an issue.
The problem with fixation is that it's controlled by turbulence.
Good, well, I'm glad I didn't say that. I think that
Memorial Day weekend, they're closed again. And the data are there, you know, down there. They're QAQC. I can look up the nitrate concentration and drive them on my cell phone at any time. It's pretty awesome. But how do we use that? Yeah, they're there. Yeah. For all of everything, or the modeling efforts that Dave was doing. Oh, I love the idea of putting them back. Yeah.
We'll get started in a couple of minutes. Okay, I think we're ready to go. If you guys could take your seats. Great. Hang on, we're just going to call the panel member who's at home. Okay, Janice, I think we should, while we're calling, you can get him.
All right. Welcome back, everyone. We're going to get started, and our first speaker is Erwin. Fill in the rest of it for me. From the Bureau of Reclamation. He's done nitrogen or nutrient studies for a number of years now. So thanks, Erwin.
Thanks, Janice, for inviting me to this.
You have reached the voicemail box of 713.
Okay, thanks, Janice, for letting me speak to this distinguished panel. I think if knowledge about phytoplankton production had mass, the floors would be buckling right now. So I'm going to talk a bit about some of the work that has been being conducted in the North Delta and the Yola Bypass to address what I'm told is like the key question that the panel is considering, and that's how this nutrient load reduction from the wastewater treatment plant will affect how much and what kind of phytoplankton will grow downstream. And I guess the smart answer to that question is I don't know, but I'm going to present some data that I think may help us provide a more useful answer anyway. So just to give you some sense of the system, the North Delta consists of basically the Sacramento River, this Cache Slough complex here, and then the Sacramento Deepwater Ship Channel, which runs parallel to the river in this direction. So, so I've done like a schematic on the right-hand side to just kind of simplify things a little bit. So we have the Sacramento River coming down and then towards Suisun Bay. Under flood flow conditions, you have the Fremont Weir, which conveys water flood flow across the Yolo bypass, which is this flood control easement. As you go downstream, you have the wastewater effluent that's discharged into the river. And what I've marked here is just two reference points. Here's the discharge point, and here's the Rio Vista, which is sort of like a master station at the bottom of the river near the confluence of the Cache Slough complex and the Sacramento River. Let's see. So another part of the system here is this tow drain that receives ag drainage and flow from some of the tributaries that come off the Cascade, the coastal range. And in this part of the system, we have effluent that's coming from the Woodland and Davis wastewater treatment plants. So, so this both parts of these systems are getting effluent. And then finally, we have the ship channel, which is basically a big, long, wide, deep canal that provides transportation corridor for ships up to the port of West Sac. And it is currently not connected to the river. It has a lock system and a flood control gate that prevents any flow from going from the Sacramento River to the ship channel. So it's a big, long, isolated slough. So those are the three parts that I'm going to talk about. So starting with the tow drain in this part that's feeding the Cache Slough complex, I just want to show basically the effect of the wastewater treatment plant, the effluent's coming in up here. And so this station is giving you the sort of what's coming in from the ag drainage, high EC, low ammonium, moderate nitrate, phosphate relatively high, NDP is very low, chlorophyll is high. Then you get into the wastewater treatment plant, still high EC. They don't denitrify, so you got very high nitrate, high phosphorus. NDP goes up, but it's still low, and then high chlorophyll still. And then as you move downstream, you get more and more of this influence of the Sacramento River water, which has low EC and has very low chlorophyll concentrations as you get way down here. So what I'm going to show now, let me go back here. So I'm going to show next is this a transect that starts here and then goes up Liberty Island. So we're going from here up north, and you can see the drawdown in nitrogen, inorganic nitrogen, during September and October. There were 3 transects performed. So as you go upstream, DN goes down to 0 in some months. So you have quite a drawdown, increase in chlorophyll concentration, but still not that high. So it indicates that there's something else that's drawing down the nitrogen besides phytoplankton growth. And certainly one possibility is the uptake by aquatic vegetation. So this is a transect that started down at the southern end of Liberty Island, like the one I just showed, and then ends up in the Tule Marsh at the northern end. And it shows the progression of just how much of phosphate and ammonium and nitrate is drawn down, how much lower it is in the Tule Marsh than in the southern part of Liberty Island. So cache is important because it's what's considered like a donor habitat. It's providing phytoplankton to the main stem river. I meant to put a slide in here from a model that Jim Cloran did in a paper in 2007 where he talks about shallow water habitats that have enough light and residence time to produce phytoplankton that is then exported to deeper light-limited habitats. So the deep system in this case would be the river. And then in exchange, the shallow habitat receives nitrogen from the deeper system because the deeper system can't use up all the nitrate. So I think that slide's later. I put this together early this morning, so I apologize for that. But the idea is basically to show, in this case, this is an event that happened in fall of 2012, where instead of the usual routing of ag drainage from diversions up in the northern part of the Sacramento River, it goes into agriculture, then it drains into Colusa Basin drain. And normally it's discharged at night landing back into the main stem, and then it travels maybe 3 or 4 days to Rio Vista, so it's a very short transit time. But in 2012, they were working on the gates that do this discharge here, so the water was routed down the tow drain instead. And what it did was, so here's the flow that went down the tow drain in this early part of the fall, and like 3 weeks later, it produced this big bloom at Rio Vista. And this has happened in other years, but there's a threshold. It has to be certain. It can't be just a small amount of flow. It has to be a substantial amount of net flow coming down the tow drain to produce this effect in the main stem. And I think a clear example is is what happened this spring, in the spring of 2016. We sampled it four times on the hydrograph. So here's in red is the hydrograph for Lisbon, which is the inflow to the tow drain, this Cache complex here. So we sampled it at the beginning, right at the peak of it, and then 2 times after the flow declined. And so you can see it starting to light up in early March. Then in early April, it's pretty hot. And then by the 28th, it's still hot and exporting biomass downstream. And by June 7th, we're starting to slow down here. But we got another bloom later. So as you'll find out later, that it's been a very productive year for this system. So Cache slough complex, donor habitat. That's kind of the bottom line of that. So the third part of the system I'd like to talk about is the Sacramento Ship Channel. We've been studying since 2012, and we do a transect that starts at Antioch down here, and then runs 60 kilometers up to the port of West Sacramento. And we also do a loop through the Cache slough complex. And we have like a dozen stations where we take grab samples and do vertical transects and zooplankton tows and all the rest. So basically the system, what I'd like to do is kind of compare this to the tow drains figures that I just showed, point out some of the similarities and differences. One of the big differences is that, as I said, it's isolated. It doesn't have any net flow. Also, it's much, much wider. It's like 200 meters wide and 11 meters deep, whereas the tow drain is, I don't know, 20 meters wide, 30 meters, and maybe the maximum depth is like 4 or 5 meters. So much bigger system and very different hydraulics. So as I said, the uppermost part of the system is pretty much isolated. There's a little bit of flow depending on the tides coming in through the gates here. And in very wet years, there is some discharge down here through these culverts. But in the whole time we've been studying it, they've never, except in 2011, was the only time I've seen those actually discharging anything. So it has a very long residence time. And so it wouldn't surprise then that it has a similar EC gradient to what we saw in the tow drain. Except it's because of evaporation, because there's no salty water coming in. So there's no ag drainage coming in to make it salty or wastewater effluent. It's just evaporation. And we verified that with isotope work. So we have this longitudinal gradient as you go from the main stem upstream to the Port of West Sac. And you have this, basically this is sort of the limit, upper limit of the tidal excursion. And then you have this isolated area. The top 15 kilometers is pretty isolated. And then you have this sort of mixing zone in between. Ammonium. So the main source of ammonium to the system, of course, is the wastewater treatment plant. It's Sacramento. So you can see its signal in the lower part where the tidal excursion extends, but there's essentially no ammonium upstream of there. Similarly with nitrate, we have fairly high concentrations because of the influx from the main stem in the lower section. But in the upper part, it often goes to zero. So it's very, very low nitrate at the top end. Whereas phosphate tends to be higher as you go upstream. So, and we've never, except for a few instances at the very top of the system, it very rarely drops to below detection. So the N to P ratio, as you might imagine, is very low in the upper part of the system. So it's very strong indication of nitrogen limited conditions in the top of the system. So that's similar to what we saw in the upper part of the tow drain system. So now the chlorophyll, as you might have guessed, the chlorophyll concentrations tend to be really, really high in the top. Well, not really, really high, but comparable to what you see at the top of the tow drain. And compared to what you see normally in the lower part of this, in the main stem of the river. Here's the fall bloom that I talked about in 2012. And below that, I'm showing the nitrate concentration to demonstrate the negative correlation between the high phytoplankton and the low nitrogen concentration. So it's another indication of uptake by phytoplankton. You can see it also here, very low nitrate in the main stem during this big bloom here. So now I'm just showing, I'm comparing the results from the early April and that late April spring bloom of this year and showing the longitudinal variation in the chlorophyll concentration. So in April, or in the blue, you see very high levels in Prospect Slough, which is in that cache slough complex. And then you have these main stem stations that were responding to that high donor condition in the cache slough complex. And then very high chlorophyll concentrations as you move up into that more isolated part of the system. And similar pattern in April, the late April, and sort of an inverted pattern for nitrate. It was drawn down to, limiting levels. So here's like 300. So here's the relationship between growth rate and extracellular nitrogen concentration. And you can see that we're in rate limited territory here when we're in Prospect Slough and even in most of the ship channel. In April, nitrate dropped to zero at these upper stations. In terms of what kind of algae are growing, this is again data for the April bloom. Got 5 minutes. So let me stress two things. One is that the phytoplankton that was produced in this donor area, the Prospect Slough, and the main stem consisted of this Allococyra granulata, formerly known as Melicyra, I think. And whereas as you move upstream, you get into much more, a community dominated more by Rhodomonas and other flagellates. So you go from basically diatoms to a greater percentage of cryptophytes and some chlorophytes. We do see cyanobacteria in the lower part, and we see microcystis in these lower stations of the ship channel because of the excursion, the exchange with delta. But in the upper part, we never see microcystis. We do, however, in the summer and fall, see some Anabina and Phanezomenon, so, which kind of makes sense because there's no nitrogen up there. So if you want to be a cyano up there, you need to fix nitrogen. So. This shows the kinds of diatoms at the 3 locations. So during the spring bloom, so in the lower station we have dominance by Allococyra, hardly any Thalassiocyra. In this lower part of the ship channel that's still within excursion with the delta, you have still a lot of Allococyra, but we're picking up Thalassiocyra. Whereas in the main of the ship channel proper, there's no more Allococyra and it's all Thalassiocyra. Zooplankton, as you might expect, high zooplankton biomass in the upper part of the system where the chlorophyll concentration is. So we have high zooplankton here, corresponding with high chlorophyll in general. Much lower levels as you go down towards the main stem. I think I'll skip that one. In terms of the composition of the zooplankton, as you move up to more lentic-like conditions near the port, you pick up, well, the copepods dominate pretty much everywhere, but as you get closer to the port, you start picking up way more Cladothera. So again, it's consistent with the idea that you're moving towards more and more lake-like conditions as you move upstream. We have strong indication that because of this negative correlation between the increase in zooplankton, we've got a decrease in phytoplankton. So we have strong indication that there's a lot of grazing going on. We did some calculations and the zooplankton can pretty much clear the water column every couple of days. Like
couple of minutes, Erwin.
So the reason we're interested in this, of course, is that this whole area is a relatively abundant place for catching delta smelt. So we're also very interested in this whole food supply angle. That's why we're concerned about nutrients, because of the potential effect on food supply. And one of the things we'd like to do, perhaps as an experiment, would be to do some nutrient addition experiments in the middle part of the ship channel, where we'd add some nutrients and then document what happens as a result. We've done it in the Delta Mendota Canal successfully, so we'd like to try it here, just to kind of verify that nitrogen, the system is sensitive to nitrogen. We'd also like to experiment with reintroducing flow downstream, just a small amount of flow. And just to end on a hopeful note, we have 2011, this is this dismal decline in delta smelt, but in 2011, which was also a year of high productivity, phytoplankton production, we did see an uptick in delta smelt, so I think there's some hope there. Finally, we were asked to make predictions. At least the email I got from Janice said that you wanted us to make predictions, so I'm making some predictions. One is that this imminent reduction in nitrogen loading from the wastewater plant will, of course, reduce the concentration of inorganic nitrogen. That will, in turn, I think this reduction will be increased by the expansion of wetland vegetation. The lower DN will reduce phytoplankton production, which will in turn reduce the zooplankton production, and in turn reduce growth of delta smelt and other native fishes that eat zooplankton, which is pretty much all fish in their early life stages. So I think since we're going to reduce the amount of nitrogen loading to the system. Right now, my understanding is that the permit requires denitrification because they have that capability, which would pretty much reduce the nitrogen supply to a nitrogen-limited system by something like 50%, I think. So, so I think that there's an opportunity to do some experimentation by not necessarily denitrifying, leave the nitrate in and load it to the delta, allow it to process the nitrate. So those are the kind of predictions I'm willing to stick my neck out on. I put two more that I'm really not as confident about because it's basically just based on what I've read, over the years. And one is that lowering, if we do denitrify and cut the nitrogen loading in half, basically we're going to further reduce the N to P ratio, which is already very low, and which could produce nitrogen fixing bacteria. And then finally, the reduction of ammonium, not sure, but my understanding from Peggy's and other people's work is that microcystis prefers ammonium or other reduced forms. So getting rid of the ammonium without necessarily getting rid of all the nitrate would tend to reduce these microcystis blooms. Okay.
Okay, thank you. Questions from you guys?
Thanks.
I have one. Irvin. Do you have any idea what the interaction of light limitation with nutrient limitation might be in this system? And also just the issue of residence time.
Yeah, that's a great question. Well, again, I kind of wish, I think I still have it in here. Yeah, so this is Jim Clorn's model with the shallow habitat, which is the donor. So it has enough light and it has enough time. And it's exchanging phytoplankton and zooplankton with this deep habitat. And in his model, it's strictly driven by dispersive transport. It's just driven by the difference in concentration between the two habitats. But as I've shown, that we really need to have a net flow to make this work. So it's not quite as simple as Jim's model. And it's, he said it's a simple model, and he does talk about the importance of net flow. So it's first step. But the basic concept, I think, is the exchange of phytoplankton from the shallow to the deep, and the loading of nutrients from the deep to the shallow. So that's the nature of that interaction. The lower part of the Cache Slough complex, so the Liberty Island, that big piece of real estate there that's a flooded former ag field, exchanges I think it's like 2/3 of its volume, like twice a day, I think, something like that. I mean, it's really rapidly mixed. So that gets to your residence time question. You don't build up chlorophyll until you get into those upper parts of the systems, both in the ship channel, in the tow drain, and in Liberty Island itself. So it's those three things. You need enough light, enough time. Where you have enough light and time, you draw down the nitrogen first. There's enough phosphate to go, So that's kind of the basis of the prediction, that if you reduce nitrogen loading to the system and the concentration of this nutrient donor habitat to the places where you have enough time and light to process the nitrogen, you're going to generate an overall decrease in the productivity of the system as a whole.
What's next? What do you have planned in the next few years?
Well, if I can get an MPDS permit from the regional board, I'd love to do a nutrient addition experiment. So there'd be a whole system scale experiment. So we get around some of the problems of using carboys and other bioassay methods. We found, I don't know if you had a chance to read the experiment, the report I did on the experiments we did in the DMC, but the Delta Mendota Canal. But basically we'd be creating a kilometer-long plug of enriched water and then just monitor it, very intensively as it kind of sloshes back and forth. And we could document changes in the amount of habitat, chlorophyll, and the type of phytoplankton. So in one experiment we do nitrate, and another one we could do ammonium, to see if that, increased microcystis, for example.
Any indication of any contaminants, or have you looked at those
Did some
possibilities?
bioassay stuff just to confirm that, the low chlorophyll in that part of the system wasn't due to some herbicide or something. So, and others have done work too on that. So, and it doesn't seem to be an issue. So,
Did you say it was or was not?
not.
Not, yeah, okay.
Okay, thank you very much.
Thank you.
Great. Next speaker.
So our next speaker is Liz Stumpner, who recently joined the USGS, and no?
Not true.
Okay, I'm sorry. I'll let her introduce herself.
I wish I could claim that, but I'm with the USGS Biogeochemistry Group here in Sacramento. Thank you for having me. I am one of many authors on this study that took place in May of 2014. It was funded by the state and federal contractors, water agency. And this study is under review currently in estuaries and coasts. This is an animation that Tamara showed earlier. And Christine, if you don't mind queuing it up. We chose to evaluate what drives the spatial distribution of phytoplankton in the North Delta. And we thought that we could execute a landscape scale experiment because the Sacramento River provides high ammonium source water to the Liberty Island complex. That high ammonium source water enters the system from the south, from the more seaward direction, and is mixed across the system. But as you can see from this particle tracking model, there is a lot more mixing in the south versus the north. And this animation was created for Tamara's study, so that's why you're seeing the same change of colors. But I just want you to focus on the more mixing in the seaward portion of the study area and less mixing to the north or landward. And the sloshing back and forth creates different gradients in water residence time and the nutrient field, as well as light. Particles are settling out over time, so possibly there's more favorable conditions for a deeper euphotic zone. Also, the Liberty Island complex has a higher concentration of tidal wetlands than in other areas in the Delta, which may be beneficial for increased rates of denitrification. And some of my co-authors wanted me to point out that the ammonium paradox hypothesis has been tested, thoroughly along stretches of rivers and channels, these deeper environments, and it really hasn't been evaluated in a system like this. So I wanted to pass that on. So the experimental goals of this study were to evaluate the phytoplankton spatial distribution and the community composition. And to evaluate that, we wanted to focus on 4 factors: nutrients, light, contaminants, and residence time. We used several approaches to measure the spatial distribution of phytoplankton over different scales. We used our in situ high-resolution mapping capabilities to cover the system in a period of a few hours, which is really relevant in the Liberty Island complex because it's very hydrologically complex. We used discrete sampling, those are represented by the orange symbols, to sample over one day in May 23rd of 2014. And from those samples, we gathered chlorophyll concentration, size fractionation. We sampled for accessory pigments, picocyanobacteria cell counts, nutrient concentrations, and nutrient uptake. We also completed a parcel tracking experiment on the second day of our field experiment. So using a Lagrangian approach, we tracked 3 distinct parcels with different starting conditions of ammonium concentration, and those are represented by the yellow and green triangles. Water for enclosures was also collected at the start of these parcels, and today I will be showing you
results from those enclosures that represent the high and low ammonium-sourced water. So what I would like to do from here is present the nutrient data. Here we are at the nutrient workshop, so I'll get that out of the way. And then I will dive into the phytoplankton abundant spatial distribution data, and then follow up with how light, contaminants, and residence time may be driving that spatial variability of phytoplankton. Here is the spatial distribution of nitrate in the system that we measured on the first day of the study. And our group is out in the system quite a bit, and we see these persistent gradients of nitrate. We see high concentrations of it pretty much everywhere. It's greater than 10 micromolars everywhere, but we're seeing some sort of drawdown of it in the very north. That is in the stair-step region. And when we compare our high-resolution mapping results to our synoptic results, we see the same pattern. So you're going to see a lot of figures like this. These are our synoptic samples that were measured throughout the system, and they're in order from seaward to landward along the x-axis. And then nitrate is represented in blue, ammonium in orange, nitrite in red. And what I'd like you to take from this figure is that there is a decrease in ammonium as we're moving seaward to landward. And in some cases, a decrease in nitrate. But you really see that pattern. It's more obvious in the high-resolution mapping data. There is a subsequent chlorophyll increase. And this is the parcel tracking results showing a similar trend. Parcel C represents the more seaward, higher ammonium source water, and there's higher ammonium there, obviously, and then with landward transport, that ammonium is drawn down. We completed biomass-specific ammonium and nitrate uptake rate calculations. These are potential uptake rate calculations, and the ammonium uptake dominated the system, but in some of the landward stations, we did see increased access to nitrate uptake or potential access, but this only represented 50 to 60% of the dissolved inorganic nitrogen pool, which, as I understand it, I'm not a phytoplankton ecologist, but that's relatively low. And we were expecting something higher, maybe upwards of 80 to 90%, but that wasn't the case. Did I miss something? Nope. Okay. And then here I just have the N to P ratio, and just want to point out that we're seeing a higher ratio between N to P in the seaward region of the area, of the study area, and a 5-to-1 ratio in the landward reaches of the system. So this speaks to the ecological stoichiometry hypothesis that we're talking about today. Now getting into the phytoplankton spatial distribution, you'll see another map of total chlorophyll abundance, and the highest concentrations are in the north. And when we pair that against our synoptic data, we're seeing similar trends, but what is striking from this figure is that the system is dominated by smaller cells. The chlorophyll representing 0.7 to 5 microns in size are in dark green, and larger cells greater than 5 microns are in light green. So the system appears to be dominated at this time on this day in 2014 by small cells. And the same is true for the parcels. We're seeing an abundance of smaller cells. The direct picocyanobacteria cell counts gave us another surprise. The cell counts are here represented as the bars, and then the red line represents the percent of the total chlorophyll A pool that the picocyanobacteria make up. And we're seeing a steady increase in cell count as we move in the landward direction. And we're also seeing that there's a higher proportion of total chlorophyll pool made up by that picocyanobacteria. And the same is true again for the parcels. Getting into the pigment data, the diatoms are represented in blue, and they're showing a rather underwhelming contribution. As I understand it, diatoms have been shown to be a lot more prevalent in the estuary. They often overwhelm the other species by orders of magnitude, or other taxa by orders of magnitude, but that wasn't the case here. Diatoms only made up to 30% of community composition. This just represents the 4 major taxa that were sampled. And cryptophytes, cyanobacteria, and chlorophytes are also represented in this graphic. So we're not really seeing a significant proportion of diatoms in the landward reaches where there should be more increased access to nitrate. So the 2 takeaways from the nutrient data were that we didn't find the large cells that we expected, and we also saw a higher proportion of cyanobacteria in the landward reaches of the system where we didn't expect them. There's less ammonium up there, so we didn't think that would be favorable necessary for the cyanobacteria community. So I've covered nutrients, I've covered the phytoplankton spatial distribution. Now I want to get into what we believe might be one of the larger drivers to the spatial distribution of phytoplankton, and that's residence time in the system. Our group has the capability, we've added a water isotope analyzer to our arsenal of water quality sampling equipment that we can head out in the system and collect a lot of data every 1 second. We're out there, and we have the ability to calculate residence time from this data set. This map was collected under similar hydrologic conditions as our Liberty Lag, as the May 2014 data set. And we calculated that residence time can vary between 0 to 60 days in the system. And this map is just showing a higher evaporative enrichment to the north along the stair step. How much time do I have? Okay, great. Okay, great. Terrific, thank you. Okay, so we're seeing higher evaporative enrichment to the north. And when we evaluate this map in terms of what we're seeing for nitrate and total chlorophyll abundance, we're seeing some things that make a lot of sense to us. Our total chlorophyll map in green is showing higher concentrations of chlorophyll where residence time is higher, but we're also seeing a drawdown of nitrate. Less nitrate is occurring where total chlorophyll is highest. And we believe this is because there is a drawdown of nitrate with an increased, the population of total chlorophyll is higher. Up there, they're utilizing nutrients. So this is looking favorable. And I guess I want to point out, when we're thinking about our results in terms of the ammonium paradox, well, maybe this lines up well with it to some extent, because we are seeing an abundance of chlorophyll in this area of lower ammonium. But as you recall from our phytoplankton spatial distribution data, we're not really seeing the favorable phytoplankton that we would expect. So these sharp gradients of total chlorophyll and nitrate align well with our residence time map. And these are persistent gradients that our group sees very frequently in this area. And we want to point out, that it's really likely this higher residence time is the cause of poor exchange of, or is attributed to this poor exchange of water in the system. Under low flow conditions, when the Yolo bypass isn't flooded, there's a lot of stagnation up in the northern reaches of the system, especially up around the stair step where total chlorophyll seem to be highest. Whenever water is stagnant, there may be increased pressure on the phytoplankton community by grazers or by like zooplankton or clams. Clams have been found in the area. I also want to point out that, and I shouldn't use the word stagnant because there is evidence of integrated mixing occurring, but there's no evidence of stratification, I should point out. But whenever water isn't moving around, there's likely an increase in temperature. And this is something we speak to in our paper that I understand the panel has access to. And if I have time, I have a slide at the end of this presentation to show you the temperature map. But anytime you're showing a map of temperature, it's confounded by time of day. So that's something that we try to be careful with when we're explaining our results in terms of temperature. Moving on to our light results, we're seeing that we used our turbidity measurements to calculate the depth of the euphotic zone, and we found that light was limiting landward in the study area. When we took these euphotic zone calculations and plugged them into a gross primary productivity model, we found that the highest production should be along the western and southern reaches of the study area, but this wasn't the case. If you recall from that chlorophyll total abundance spatial map, spatial distribution, we found the highest production is in fact in the north. So this is our evidence suggesting that light isn't necessarily controlling spatial variability. I spoke earlier about those enclosures, and enclosures are something that has been talked about a lot today, and these are really useful because, and especially when you pair them with field experiments, you want to see the same results. Why would something be happening in an enclosure and not in the field? And certainly there are reasons why those results don't always match, but we wanted to at least test whether diatoms and some of these larger cells could grow in the presence of higher ammonium water versus lower ammonium water. So this graphic is representing the higher ammonium water concentration. Yes. And you're seeing ammonium in orange, and that is the dashed line that is drawing down when ammonium reaches, around 1 micromolar. Nitrate is available, and there's a subsequent increase in the chlorophyll community. Those larger cells are represented by the light green color. So this enclosure in particular is growing larger cells. It's also growing diatoms. Those pie charts at the top right of the screen, the blue color represents the diatoms, the purple color represents the cryptophytes. So it does appear that this enclosure is growing diatoms. And when I overlay, the results of the low ammonium enclosure, it's a little tricky, don't get dizzy, but I didn't really know a better way to do this. You're going to see that drawdown. Well, that ammonium concentration is low. This is the low ammonium enclosure. But you're seeing a steady decrease in nitrate, and with that, a bump in chlorophyll. Around hour 70 of the incubation, chlorophyll starts declining. We believe this is due to nutrient limitation. But the point of this slide is just to show that both enclosures grew diatoms, despite starting ammonium conditions. So we're taking this at face value to say there's nothing in the water, contaminant-wise, whether that's ammonium or possibly pesticide or trace metal concentrations that would inhibit growth. We did measure trace metals. We did measure pesticides. I'm not presenting the trace metals today because that was not significant, but I do want to bring up our pesticide results. 18 pesticides were detected in our synoptic samples and on starting parcel water for the parcel tracking experiment. None of the samples with EPA benchmarks for toxicity for non-vascular plants exceeded the EPA standards. But I should point out along the bottom here, there are 5 compounds with asterisks that do not have benchmarks for those non-vascular plants. Malathion, which is an organophosphate, it's an insecticide. It exceeded invertebrate toxicity in 3 of the 11 samples. The insecticides are presented here on this bar graph in yellow, and the malathion was abundant at Lindsay Slough, Upper Cache Slough, and Haas Slough. The chlorophyll concentration of all these sites samples are also are the numbers on top of the bar graph. So this Haas Slough sample with the highest combined concentration of total pesticides actually had the second highest concentration of chlorophyll. So we don't believe that our pesticide distribution is necessarily aligning with our phytoplankton spatial distribution results. In conclusion, we found a lack of large cells despite low ammonium. We found higher cyanobacteria in areas of the system where low ammonium was present. And we didn't feel that, our results show that our phytoplankton distribution didn't really respond to increased access to nitrate in the system. We think that residence time is playing a large role in the phytoplankton spatial distribution. So we're not ready to say that it's nutrients alone. We think residence time is playing a large factor, but our light and contaminant data didn't necessarily assist with explaining our results. So what does this mean? I'm not going to necessarily give recommendations, but this study in particular, and many that we see this afternoon, possibly will assist with narrowing the gap between these controlled incubations or lab-based cultures, which are all really relevant. But I'm a field junkie, and I work with a bunch of other people who are. So we'd like to try and maybe make this happen on larger scales. Doing the landscape scale experiments might be really cool. We don't know that lowering ammonium alone with the upgrade to the wastewater treatment facility is going to change the system. The hydrology of the system most likely plays a large role. And as far as the effects of restoration, as you all know, a lot of restoration efforts are taking place in the system. So how are those restoration efforts going to affect distribution of phytoplankton?
Okay, thank you very much. So we have time for some questions before our afternoon break.
I have a question about your contaminants and pesticides. So I understand from talking to people here that this is quite seasonally oriented. So at certain times of the year, you might have a flush of pesticides in, but not at other times. So in terms of your work, where did your timing relate to the flush of pesticides?
One of our co-authors, Jim Orlando, he's in the pesticide fate and research group. And I know, I can't speak for all of the compounds, but I know that some of the insecticides, and anyone else can help me here that is more familiar with spraying schedules, but that organophosphate malathion, he thought it likely was like applied, recently. He said that I don't know if it's appropriate to say half-life, but a lot of those compounds degrade quickly, and the concentrations we saw in the northwestern section of our study sort of lead us to believe that that was applied like days before we were up there. As far as herbicides, I don't know like the spraying schedule that I don't even remember the name of the agency. Boating and waterways. I don't know how frequently they spray aquatic vegetation. Diuron is one compound that's received a lot of attention. We didn't see a very strong, we saw a very weak relationship between that compound and other herbicides with the chlorophyll results. But I'm sure that timing does play a large role.
So in general, their half-lives are quite short, and therefore it's likely to be a short-lived effect if there was one.
Yes, Steph.
Please.
Come on up.
Hi, I'm Stephanie Fong with the State and Federal Contractors Water Agency, and typically malathion is something with a very short half-life, so Liz is very correct, but what we find in our system is the majority of the contaminants come down through storm events, and this wasn't meant to capture storm events, so timing is a bit off for them.
With regard to contaminants, there are various terms that are used in the reading that we've been doing or hearing: contaminants, toxins, pesticides, herbicides. How do you evaluate currently the robustness of what is known about their effects in this system? Does there need to be, in short, does there need to be more? Because it seems it's coming across as sort of a topic that's out there as a possibility, but not necessarily enough information to even draw any conclusions at this point as a relevant factor.
There's a manuscript that's hopefully coming out in December that explores a lot of the effects that we see from the contaminants in our system. And there have been quite a few, particularly more on the insecticides than the herbicides.
Okay.
So we definitely have a huge lack of knowledge on effect concentrations of any of the herbicides as well as the fungicides that are readily applied and quite liberally applied because they also provide a beneficial effect other than the pest management. A lot of them are actually going to increase yield, crop yield, so they are applied pretty regularly.
So one more question following up on that. So this is totally out of my area, but I'm just curious. So is it true, or is it a general statement then that the zooplankton, the animal communities may be more sensitive than the phytoplankton? Is that right?
That may be the case, but like I said, we definitely have a large gap in what the phytoplankton community is seeing effect concentration-wise to a lot of the chemicals that are being detected. Jim has found tons and tons of herbicides and fungicides that just don't have any effect concentration data at all.
So this would be a knowledge gap going forward.
Yeah.
I want to point out, and I don't know how frequently he goes out, but Jim and his group do head out to Liberty, I think like at least every couple months. I don't know when, he publishes a lot of USGS reports. I don't know that he has one out on that yet, but the data is getting collected.
So unless he has more funding coming from another source other than us, I think his monitoring is going to be very short-lived.
And Antonietta, I want to make sure that you're on the phone and you have a chance to ask any questions if you have any.
She just texted me a question, which I'll toss out there.
Great.
For others. She's heard various people mention the potential effects of restoration, and I glossed over so many things in my introduction, but her question was, why mention the effects of restoration in the yellow bypass area? Do we expect that it's going to have a big effect on either phytoplankton or the nutrients. So,
I'll take a stab at it. Well, I just know with a lot of the restoration work currently underway, the idea is that restoration of tidal wetlands slows down water. And there's a fine balance between depth of the water column, the speed water moves through the system, and how that relates to nitrification rates and the ability of the food web to respond under favorable conditions. It's like there needs to be the perfect storm. That's not the right phrase, but the perfect set of conditions need to be met. And for some time, many people believe that restoration of tidal wetlands will achieve that. But there are, I mean, this is just kind of this is like the bad news that in the system of tidal wetlands, we're not seeing the response we want necessarily. But this is just over 2 days and happened at the end of a large drought. And water temperatures were above 18 degrees Celsius. So I think that's why we're kind of spinning this in terms of restoration, because there's just a lot of unknowns associated with it.
I was just wondering if those restoration efforts are designed to increase the water clarity, so using the wetland areas as a way to trap sediments and things like that, and therefore increase water clarity. Is that part of the goal, or is the idea that they're going to trap nutrients? What's the goal, and how does it relate?
In a system with a higher concentration of tidal wetlands, you would expect more sediment trapping, clearer water, but we found a lot of light limitation around areas where tidal wetlands were more prevalent. And conditions, Jim Klern has discussed a lot about the value of how depth, euphotic zone depth plays a role in different habitats as far as creating more favorable habitat for lower food web. So we would think that the abundance of tidal wetlands would assist with that.
Okay, any more?
Yeah, one quick question back to the contaminants. So the San Joaquin River area, is that more susceptible to contaminants than your area?
There's a lot of agricultural activity surrounding the North Delta, so I don't know where it compares to San Joaquin. More ag in the San Joaquin.
Okay. Okay, so let's take our break. We'll come back at 2:40. We'll have 2 presentations and then a little more than an hour for open discussion, and we'll make sure that we have time for the audience to ask whatever questions they have or make comments. So come back at 2:40.
It's really good. Not much. And there's different types of agriculture. Pretty low. Yeah, there's a raise.
I did.
Okay, I got an email.
My classes have my They said, they were sitting on my desk, and so they said, somebody's voice they said was songs. My answer was that because it was actually
We've got just a couple more minutes. Okay, let's get going again. If we could take our seats and have our speaker come forward. Is she? Oh. No. Okay, Jan. Might put it up on. Okay, we're ready to go. Well, she's right there. She's talking to Dave. If worst comes to worst, we'll run through the slides. Alrighty, the last session of the day. We have 2 more talks. Antonietta, are you still there?
Yes, I am.
Thank you. Great. Okay. We had our speaker. Oh, great. Okay. You were below the podium.
Hi, I'm Jan Thompson. I work with the USGS in Menlo Park. I was asked to discuss the clam part of the story. So I'm going to be doing that. Depending on how much time we have, I may end with a discussion of a recent paper we published on how clams may select for consuming diatoms over non-diatoms in this paper that we just published. Okay, instructions probably meant I should have gotten, okay. I've included a couple names on here. I did check with them because an awful lot of this work is in conjunction with Lisa Lucas and Wim Himmer, and I want to acknowledge my co-authors certainly. So this is sort of an outline of what I'm going to talk about. I have a number of slides in here with information for the panel, figuring that you don't really want to go through the literature and find out some of these numbers. And I'm not going to cover them all in the talk. So when I skip over some of these, this is because I think they might need them. I'm going to talk, I say a very little about bivalve ecology biology. That's not why we're here. I'm going to talk about the distribution of both bivalves. We have 2 bivalves in the system that can limit phytoplankton. I'm going to talk about 2 methods. The first method is the simplest method, which is to compare time series of benthic grazers and time series of chlorophyll. Do they match? And the answer is, with Pitamacorbula, the estuarine clam, yes. However, that doesn't answer all of the questions, because other things could cause that. That can be cross-correlates. So one of the things that women I've done is to look at the mass balance question. Look at secondary production, estimate how much carbon these guys are consuming to grow that much, and then looking at primary production and saying, does it make sense? Is there enough food for them, or do we think they're eating 100% and they're actually only eating 25%? Basic question. Then if I have time, I'll talk about the simple models. So the top plot, I think most people have seen. This is Pitamacorbula that invaded Suisun Bay in the fall of 1986. This is the chlorophyll trace. And this is Pitamacorbula. It's an estuarine clam that lives throughout the bay and up to 0 salinity. This clam lives in this part of the system, all the way through South Bay, by the way. And Corbicula, which is a freshwater clam, lives in the delta. And we also have examples where in Frank's Tract, where we have a lot of Corbicula, we don't have chlorophyll. In fact, we can consider it a net sink for phytoplankton. Mildred Island, at the time we did this study, had no Corbicula in it, and we considered it a net source for phytoplankton based on our analysis. So this is one of those tables. I'm only going to highlight a couple things, and you guys can look at the rest if you want. Big difference here is Pitamacorbula came in 86. Corbicula came in the 1940s, which was alluded to earlier. So we don't have those wonderful time series of what happened when Corbicula came in, because nobody was studying. They're both opportunistic species, and they have an affinity for disturbance. This is really bad news for restoration. It means if you disturb the environment, they both move in very quickly. They filter feed on phytoplankton, bacteria, and zooplankton. So these are, if you're going to design your perfect exotic invasive species, these are pretty close to it. The difference in the two is their salinity tolerance. Pitamacorbula is an estuarine clam. Adults live actually in fresh water, right at the boundary of fresh and salt water. Corbicula live down to 10, a salinity of 10, so they overlap between 0 and 10. This is not good news. This makes it really hard to limit these guys. The only thing that really is handy is the juveniles don't overlap, but they butt up against each other, a salinity of 2. Corbicula juveniles are limited to salinities of less than 2, and Pitamacorbula greater than 2. And this is possibly the most important detail for this group. Pitamacorbula is an awesome pumper. It filters 400 liters of water per gram dry tissue at 20 degrees C. Corbicula, 100 liters of water. So it's, if you have equal amounts of biomass of both clams, Pitamacorbula is going to filter 4 times the volume of water that Corbicula does. And it's the one that moved in and we saw the biggest changes to the system. This is a plot showing you where they live. To give you a better feeling, as I said, Pitamacorbula lives all the way to the southern end of the bay and into the sloughs at the south end. We collected them when they first came in Central Bay, so they pretty much can live anywhere. So they live from a salinity of 35 to 0. Corbicula lives from 10 to 0, to fresh all the way up. This little inconvenient place that they overlap that moves back and forth, of course, with the hydrology of the years. This is the low salinity zone, which is the area where we've had the largest reduction in food supply, presumably for the fish. So this isn't, I don't know about perfect storm, but it wasn't the ideal two clams that we could have had introduced. I'm going to talk briefly about the distribution because a lot of people think that this is like 5 or 6 places that we get a lot of clams. That's not true. The reason why we know what we know about the distribution is because DWR has been doing a spatially intensive study since 2007 in May and October of every year. They take 175 stations. That's really critical. We have 10 stations that they've been taking monthly, and that data is really valuable for understanding seasonal cycles and a lot of other things, but they don't tell us what the distribution is of something like this in space. So this is 2007 to 2012, all of the station combines on one graph, just because it makes it a little easier to see. This is data from 2009, 10, and 11 combined, and this is filtration rate, which is, for those who aren't familiar with filtration rate, it's cubic meters per square, so it's cubic meters of water per square meter per day. When you do that, it comes down to meters, you can reduce it to meters per day. That's very handy because you can get to a rate that you can compare to phytoplankton growth by just normalizing it to depth. The clams only affect things on the bottom. They don't, they depend on physics to bring the food from the top to the bottom. They're only on the bottom. So in combining these 3 years, I've done that for 2 reasons. One is that 2009 was a very dry year. Therefore, we'd expect the Tamacorvula to move upstream. 2010 was a normal year. 2011 was a wet year. And then I said, okay, are there any persistent patterns that would indicate it doesn't matter what the hydrology is? This is a persistent pattern that we see. So these are, believe it or not, pie diagrams that show total filtration rate between Corbicula in orange, Tamacorvula in the blue. And here's the overlap zone. If you look carefully, you can see that the pie diagrams are real in there. There are a couple things that are incredibly persistent. One is spring in San Pablo Bay, no clams. And that is because the fall migration of birds take them out. And it is a very impressive thing. In the data, they're there one day and then they're gone. Same thing happens to a lesser degree in Grizzly Bay and Honker Bay. So these are areas where what I call, both of these areas where we can get spring bloomlets because the grazing rate's really low in spring until the clams come back in and grow and start filtering really fast. The other thing that you can see is this area right around the confluence, which is not surprising, it's basic ecology, that these two clams where they overlap is where we get the lowest filtration rates because they're both physiologically challenged. We have a flip between Tamacorvula is low in spring, high in fall. Corbicula is high in spring, lower in fall. So they have varying opposite effects in some ways on seasonal patterns. This is taking that first plot, showing when the chlorophyll declined when Corbicula, Tamacorvula moved in, and looking at the chlorophyll plots in time by season. So this is March, April, May, June, July, August, September, October, showing it's lined up with the one above. This is where the clam came in. The color is log chlorophyll, log 10 chlorophyll. And you can see before the clam came in, we got pretty consistent in these months, some very high currents of chlorophyll. What's interesting to see in this plot is it lasted, for those who haven't looked at the historical record, it lasted a long time. These are in 1971. As you can see, it lasted from May through October. So this was a really big event in North Bay that we've now reduced to essentially chlorophyll levels of 0 to 1 to maybe 3. If you look at the phytoplankton growth rate in the triangles here, the open triangles, plot the clam grazing rate, so that's take the filtration rate of meters per day divided by the depth of the water column, so we're now in the same units per day. You subtract those two, you end up with net growth that's almost always negative. That's our first hint that it's not just coincidence that those two things are lining up, but we mathematically can see that chlorophyll is negative. The chlorophyll is declining because of the clam. This is, again, the chlorophyll over that same time period and the water year. You can see there's one little eruption here in 2000, and maybe people in this room can argue with me on that. I think it's actually in the decade. So this is when we looked at the production numbers, when we looked at primary production versus secondary production, and moved that secondary production, it's not equal. How much of what we see of this clam tissue that's been gained, how much did they have to eat to get that? So that's one of the calculations that we made here. We assumed a 25% trophic assimilation rate. In other words, 1 gram of tissue that we see as growth meant they had to consume 4. So that incorporates things like respiration, reproduction, natural weight loss following reproduction. And this is really where I start to, I've been very wishy-washy for years about did they really cause the phytoplankton to decline, and this is the plot I'm no longer wishy-washy about. This was in 2006 and 2007. P is phytoplankton production, and in the greater than 5 meters, I have to always remember which way this is, in the shallow water and in the deep water, and this is the consumption by a combination of mesozooplankton, microzooplankton, respiration, and clam consumption. So clam is this green color. And in 2006, which was a very wet year, we saw the clam population really get nailed back all over North Bay in spring of 2006. And even with that, we can see that we are estimating higher consumption than we were getting in production of phytoplankton. So one of the questions we continually have, and I still have, is what are they eating? Even if you remove things like the zooplankton grazing, in many cases the clam consumption is as great as what we would get in phytoplankton production. So we have a couple things that we assume were hypotheses. One is that it is phytoplankton ejected from the rivers down into the bay. That's very possible. It's also possible that these guys can eat DOC and assimilate it. There's some increasing evidence that my comment is, if we look at any bivalve, we may find that they all can do this. We just haven't done the experiments to know if they can all do this. But if they can, that's an awesome food source in the system. And it could certainly explain this. Need to figure out the time here. Okay. We see, as I said, we don't have this nice, neat, clear story with Corbicula because we don't have these time series. But what we do have is a series of studies that the USGS did in the early 2000s, starting in 1999, where first thing that we did was we just looked at Frank's Track and we looked at Miller's and at Mildred Island. And what we found, as I said earlier, was that we have the chlorophyll increase of gradient in Mildred Island, and we have no clams, which you can see down here where the grazing rate of Corbicula is shown in Frank's Track and shown in Mildred Island. And you can see that the grazing rate is on the order of 5 to 8 meters per day in Frank's Track, and it's on the order of almost 0 in Mildred Island during these time periods. I want to comment on the fact, because Erwin just commented on Jim's model that was showing the importance of connectivity. Yeah, the grazing rate was really low on Mildred Island, but look at these sloughs connecting and around the island. Those are some of the largest populations of clams I've ever seen. So the export of phytoplankton out of the system, which we know they were exporting, maybe didn't get too far. Just a thought. Corbicula did come in 1940. I just want to make this comment because everybody assumes that now it's stable. It's not stable. We don't know what they're doing. They're spreading throughout the world right now. They're spreading in South America and parts of Europe. And every place they spread, it's a new experience for everybody. We all learn something new. This is, as I showed you earlier, what Mildred Island looked like. We didn't know why the clams were not in Mildred Island. We have all sorts of hypotheses, none of which can be proven. From contaminants to the food doesn't make it to the bottom, which makes no sense because Corbicula lives in reservoirs, very deep reservoirs, and does very well. This is what it looks like in 2007. Something happened between our last sample, which was 2004, and DWR's first sample in 2007. They moved in and became very common. So this area that we assumed was a phytoplankton, an example of how we can produce phytoplankton in the system, build a deep enough island, do whatever the magic is to keep the clams out and produce phytoplankton, no longer true. We haven't gone back in and run the experiments, but I'm guessing based on the grazing rates that I'm looking at, we may have better ways to spend our money and time to rerun those experiments. All right. So I'm not going to make, those who know me know I'm not a phytoplankton person. I do physics and I do benthic ecology, and I avoid things like making statements that phytoplankton are going to grow or not grow if we do X. So what I've done here is say, I think that we right now can say that Phytoplankton can limit phytoplankton growth and biomass in the system based on the multiple ways we've proven that in sustainability. Their reaction, if you do something to increase food, is going to be very fast. That we know based on the time series of data we have at these 10 locations that we have long-term data. They respond amazingly fast to an increase of food. However, the birds don't go away, the fish don't go away, they get taken out every spring, every fall, and they don't come back till spring. So what the upshot of that will be with Phytoplankton is they're going to persist in larger numbers in the deep water, but the shallow water may not be that affected other than the end of the production period. By late summer, early fall, they'll be back and they'll be in large numbers, but in spring, I don't think, it doesn't matter what you do, they're not going to come in. They start to reproduce in spring. It takes a while. Corbicula looks like they can limit phytoplankton in shallow water. There is a study that was done in the 80s that said that the food limited at 20 micrograms and 40 micrograms of chlorophyll per liter. I find we never get those levels in the delta, so if that were really true, they wouldn't be here. So they are capable of eating a lot of other things, so maybe that's how they're surviving, or maybe those levels need to be rechecked. And the guy who got those levels just walked in. Chris Foe, this was part of Chris Foe's thesis. Corbicula is evolving. Distribution is changing. We shouldn't assume that what we see today is what we saw 10 years ago and what we're going to see in the future. We do see them in places that you've heard discussed in the last hour or two. We see them in the Deepwater Ship Channel up to about 2/3 of the way up. We see them in Cache Creek, Cache Slough, Liberty Island, the yellow area, except in the areas that are periodically drained. That's a good way to keep them out. As I said, they're probably food limited, so they probably won't react to any increase in food. Is that it?
Great. All right. Questions? Oh, yeah. Thank you. Good job. Nice and concise. Good.
We're working on that.
Yeah. All right. Questions?
This time of day, it better be.
In some of the reading material that we have, the clam distribution is sometimes described as somewhat patchy across the
Corbicula.
benthic, yeah, okay, for corbicula, and that this may influence whether or not it correlates with phytoplankton distribution, I guess would be the best way to say it in the overlying water. My question is, does patchiness really factor into the effectiveness of these clams in clearing the water in general of phytoplankton? I guess in terms of going from 100% coverage, let's say, of the benthic bottom to 10% or something in between, is that going to correlate directly with their effectiveness in clearing the water? It seems to me that you would need a certain volume of water flow past them, or have water go past them in order to facilitate the clearance. We're hearing now a lot, obviously, well, is it clam grazing, or is it nutrients, or is it something else? It seems like that factors into their overall, if you want to call it success, in basically clearing the overlying water of phytoplankton biomass, or not.
There's a couple things that make a difference as far as their ability to clear the water column. One is the water depth. They're dependent on the turnover, the turbulence to bring the food to the bottom. If it's stratified, they have less access to food that may be growing on the surface, and that kind of those two are much larger effects than the question you asked initially, by the way. Their patchiness, if they were huge patches, would be important. If we had kilometers between patches, I would say it's close to, in most of the system, tens of meters. They do have microhabitats that they live in. Thymocorbula is much more homogeneous than corbicula. Corbicula also lives in an environment that's a little, on the bottom, steadier than the tidal environment that Thymocorbula lives on. I think that mixes them up, gets them sort of arranged. I think corbicula depend on microhabitats. So there's a couple things that happen. If we have clams from me to the monitor, somewhere in this area as the water flows over, your big brother upstream just ate it all, right? And so now you're dependent on the physics to bring it back down and how fast that is. So there are hypotheses that aren't necessary, they haven't been proven, but there's something to think about. Is the size of the clam bed a function of how fast they filter the food out of the water column in general, or in average in that spot? And therefore, in Sacramento River, you need to have X meters between patches in order to replenish the amount of food to the bottom.
So each patch can have its...
Yeah, it's a very interesting question, obviously, pure science kind of engineering question. It's fascinating.
So would it be accurate to say we don't know enough about that now in terms of how the distribution of clams in this system are?
Okay, so I get a lot of questions on what should I use for grazing rates from a lot of modelers. And the way I use grazing rates with the modelers that I work with is I give them the lower limit and the upper limit of grazing rates. And I said, we got to run both. Because at some point, and many times it doesn't make any difference. In that gone is gone, right? But if we knew what else they were eating, it would make a difference, because then we'd be worried about whatever else they were eating. But there are certainly instances where using the lowest grazing rate, which assumes they're only eating 2/3 of the time, assumes the physics is such that they're really limited in food. Those grazing rates can be an order of magnitude less than the grazing rates of the maximum that we might expect. And in some instances, with Phytoplankton in particular, those are very, very different. Corbicula, lower pumping rates. That's part of it.
How much of a control do the clams have on microzooplankton and the mesozooplankton?
Surprising, some. There's a couple papers, one by Val Green, who's a Wynn Kimmerer student, that shows that she actually has a grazing rate on microzooplankton. And my memory is that it's about 30% of what we see in phytoplankton. We know they remove them. We don't know if they assimilate them. Those studies haven't been done. Wim showed, one of the earliest studies on Phytoplankton was Wim's work showing that they removed the nauphlia from the water column very efficiently, and almost as efficiently as they removed the phytoplankton. So they're capable of doing some pretty impressive things. This is just Phytoplankton. We know that Corbicula is capable of removing rotifers. There's some recent stuff that indicates they're capable of removing some microzooplankton other than just the rotifers and the copepods. Both of them are amazingly good with bacteria. And Corbicula also deposit feeds. So they can, if they run out of food in the water column, they can take it up off the bottom in the mud, which makes them just amazing. I don't know why they didn't take over the world. They're just really amazing.
Well, they're not done yet, so maybe they will. So.
Great.
So, Jan,
one more question?
Speaking of taking over the world, do you think that they play a role in the advent of buoyant nuisance organisms like microcystis because they can avoid being filtered?
That's true of the zebra mussel, I think. Are you thinking about that work?
Well, I'm thinking of the analogy to Lake Erie, for example.
I think that our turnover rate, our physical turnover rate, is so high, I find that unlikely. You know, the reason why this works with the zebra, there's a couple reasons why this works with zebra. Zebra mussels can actually increase the population of blue greens. So one of the reasons that works is it stratifies. Our system, I mean, it stratifies at Carquinez Bridge pretty consistently. Other than that, in the shallow water, you have to really work to find stratification in the shallow water, which is where the action is. So I think probably not just based on the physics, but in Stockton deep water turning basin, where it stratifies all the time, there are no clams on it. And I think it's because it goes in oxygen. We have sampled all through the turning basin, wondering if they were there just.
But in the tracks, you think like this is possible there?
Could be possible in the weeds. In Agarria, it definitely stratifies in Agarria. You can stand in the Agarria and feel the stratification, the temperature stratification. So it's possible there, actually, and that's a really interesting question, because that is one of the areas we are seeing persistence. Great question.
Well, microcystis seems like it's learned to take advantage of almost anything.
Yeah, so together we have two.
Clams, I guess.
We have two strong organisms here.
Okay, thank you very much. All right. So we have one last presentation and then quite some time for Q&A and discussion. Our last presenter is Cliff Dahm.
Cliff Dahm is the lead scientist for the Delta Science program, and when is the paper for the Delta nutrients coming out? Okay, so we're waiting for that, and meanwhile, Cliff's giving us a preview.
Only a limited preview. Actually, I'm going to talk about a few other things too that are of interest to me relative to where we might go with this whole question. So let me start with first comment. I've been interested in nutrient biogeochemistry for about as long as Hans Perl has been interested in nutrient biogeochemistry. In fact, I took my first class in biogeochemistry, chemical oceanography class in January of 1993 or 1973, and it was interesting that one of the very first papers I read was the Richard Dugdale paper, talking a lot about the work of Richard Dugdale, which still amazes me that we are still having that discussion. I want to make a disclaimer. First of all, I have never taken a sample from the California Delta. I've never measured a sample from the California Delta. I've never measured a rate from the California Delta. So my interest and my knowledge of biogeochemistry is from other systems, and I'm going to bring some of that information actually into my discussion. I did think that Hans's introduction about the diversity of systems that he worked in, ranging from Lake Tahoe with a second disk of 35 meters to lakes in China with second disks of measurements on the range of centimeters. I can come close to you, Hans. I've worked at Crater Lake where the second disk measurements are over 40 meters. I've also worked at Mount St. Helens immediately after the eruption where second disk depths were on the order of 30 centimeters. Similar range of habitats that have been studied. Go to the, do I have control of the slides here? So I was glad that Janice this morning spent a little bit of time talking about the 2009 meetings. Two of the four members of the panel that was convened in March of 2009 are back with us, Hans Perl and Mary Kay Ward. Welcome the two new members. Pat Mulholland lost a battle with ALS, so he's no longer with us. And Joe Meyer was not available, so that's how I think we assembled this panel with two new people and two people that had a history. The 2009 March symposium or workshop generated a report that Janice has already told you a little bit about earlier this morning. She highlighted 4 of the recommendations. There were actually 12 total recommendations in that report. The 12 recommendations are here. I'm not going to go through them one by one, but I think it's useful for those of you in the audience to take a look at the suite of recommendations that the panel made. I think on a number of these recommendations, there has been very substantive progress. And a lot of what we heard today up to now really delved deeply into a few of these specific recommendations. So I really commend that panel, and I also commend the process by which we were able to get resources out to the scientists and all the work that the scientists have done. It really helped us move forward on some of these kinds of issues. So I wanted to highlight that. There are a few of these up here that I still think we have a ways to go. So what I was doing a little, in my mind, mental exercise of how many of these have we done well, how many of these have we done some, and which ones haven't we really done. A couple, 3 up there that we really haven't touched, quite honestly, at least in my opinion. And actually 2 of them that up there that we haven't done very much on, and we've heard a little bit about today, is we still have a long way to go to understand what's going on with the system. And so that would be one of the take-home messages that certainly I'm going to try to give you. So my role as the lead scientist for the Delta Science Program is to basically try to get resources out to our researchers. And so we don't have a huge budget, but we do have a modest budget. In past, we've been able to tap into bond funds, and so bond funds have been available to us. And so when we had the panel in 2009, we got the recommendations from the panel. We were able to develop a proposal solicitation package that was issued in 2010. And we were able to fund 14 projects that had a Delta-centric focus. 2 of those 14 projects really focused on nutrients. And so 2 that focused on nutrients you're seeing here is one that went to Tricia Gleibert and her colleagues to look at some of these issues of nutrient form in particular and their effects. And so many of the papers that have been talked about today have been cited today are byproduct of that award. They had 8 publications, peer-reviewed publications at the end of the award, and they've actually, I think, have 3 or 4 additional since that award. I think quite productive in terms of their output. Similarly, Alex Parker got a big project to study some of the harmful algal blooms in the system, and again, 6 publications at the end of the project, and 3 others in the press, and also wanted to acknowledge Peggy who's in the audience, who was also a leading researcher of our understanding of what's going on with microcystis. So certainly know more about microcystis now in 2016 than we did in 2009, that initial workshop. Before I go on further, I'm going to tell a little personal story. This is sort of my link back to this whole concept of the idea that forms of nutrients and ratios of nutrients can play some significant roles. And it has to do with work that I was doing in the middle Rio Grande in Mexico, which is my home. And this is 3 papers that were published by a doctoral student of mine with a statistician colleague of mine that came about because the student walked into my office one day and said, I'm married, I have a family, I can't work full-time on a PhD, but I'd really like to do a PhD. Is there a way we could do a PhD using existing data? So we went and got USGS data, which was quite substantive, confirmed with USGS that they would let us use this data, and he actually did 3 papers, all of which got published, all of which I think are very interesting papers relative to the nutrient biogeochemistry of a riverine system that starts out much like the Sacramento River, as a pretty pristine river in the headwaters in the Colorado-New Mexico border, travels down through some gorge country, out into some Pueblo country, and then finally ends up in an urban area and an agricultural area. And you can see the changes in water quality as you move down through the system, but when you look at the data, it's quite clear that the major change in water quality as you progress through the system occurred because of a large wastewater treatment plant in Albuquerque. That wastewater treatment plant at that point, during most of the time of his study, was, as the SAC regional system, discharging an overwhelmingly ammonium-dominated effluent. It was discharging it actually into a system with much higher pHs than we have here in the Sacramento River. Quite commonly, you would see pHs during the summer high photosynthesis period of close to 9. For those of you who know a little bit about the ammonia-ammonium couple, that means that a lot of the ammonium is being converted to ammonia. That third paper that Howard Purcell wrote, he's by the way, he's now a scientist at San Diego National Labs, was basically a modeling study is how often does the acute and chronic ammonia toxicity occur downstream of this wastewater treatment plant. So during low flow conditions, you did quite regularly see acute, and for many, many days a year you saw chronic. So turns out, quite by happenstance, that one of the Indian tribes were suing the city of Albuquerque for the poor quality of the effluent. The water was used ceremonially. They won that lawsuit. The plant was upgraded in 1997-98, and basically we no longer see this kind of acute or chronic toxicity related to ammonia. And the endangered fish species, which has a combination of cleaning up the water, captive breeding program and reintroduction, better understanding of the life history of the fish and getting flows when they are dispersing and spawning, or they're dispersed spawning fish. Fish is actually doing a lot better than it was 15 or 20 years ago. So there is an example of a success story relative to an endangered species that has a link to nutrients, to life history understanding, to food, and to the fundamental science done by a hardworking doctoral student. All right, a couple things I wanted to say about the Delta. The aquatic plants that are out there. We have seen the aquatic plants do very, very well during this drought. And in this past year, about a third, 30% of the open water in the Delta at the end of this recent growing season in 2016 was covered by either submerged aquatic vegetation or floating aquatic vegetation. These are data from Ted Summer and from the group at UC Davis and at DWR. So example there on the left, as you can see, Frank's track in 2014 versus 2015. So aquatic plant cover in the Delta, in the open water of the Delta, has been increasing, and this past year reached what I believe the record. So what does that mean to the primary production that goes on within the system? So this is a little thought exercise. It's not been, I've done a lot of digging deeply into this, but basically there's nice data on annual phytoplankton primary production. This is particularly the work of Jasby and King, like that. Kind of a typical number times, this being a median, values ranging kind of a factor of 4 around this median value, around 70 grams of carbon per meter squared per year. What about the two major groups of aquatic plants? Well, we have annual primary production for submerged aquatic macrophytes. Well, I said, where can I find good data on primary production of some of these aquatic plants? I popped open my Wetzel textbook. Wetzel did a lot of work on aquatic plants and has table after table after table of rates. They didn't find a lot of information on Brazilian waterweed, which is our dominant submerged, but I certainly found on a number of other submerged aquatic macrophytes. I found one study in Argentina that actually did do some work in the native habitat of Brazilian waterweed. Their value was kind of in 500 to 700 grams of carbon per meter squared per year. So somewhere in that range is kind of the production that you would probably get from the submerged aquatic macrophyte, and then the free-floating forms, the water hyacinth, always very, very high rates of production wherever it's been measured, but anywhere from 1,500 to 400 grams of carbon per square meter. What's the stoichiometry of the nutrients in those plants? I don't think we have a ton of good data on that. In fact, I think it's fairly limited. Are we looking at a carbon to nitrogen ratio of 10, 20, 30? I guess it's probably going to be in the 25-ish range, but certainly we can get those numbers. We can do the same thing for all of the aquatic plants out there. I think with a good assessment of the stoichiometry of the plants and some studies to get an idea of uptake rates and things like that, there's a way we could actually begin to address this question. I posit the hypothesis that these aquatic plants, with their densities and with their coverage that we see now, are a major source of nutrient uptake. We also have been seeing these blooms. I mean, you had some very nice presentations with lots of good data showing the blooms that we've had in 2016. Multiple blooms. Initially, I heard about the first one that occurred after the Yolo bypass flooded in March, big bloom in April, but there's been successive blooms throughout spring and summer. The last one in the summer was actually experimentally induced by Ted Summer and his group of DWR. They actually got about, I think it was 13,000 acre-feet of water from the irrigation district, routed it down through this lower bypass area, designed a very nice sampling program, sampled continuously at many of these sites, linked up with the USGS, also had the capacity to make these kind of measurements, and again produced another of these blooms. This one you can see is in August, the latter part of August. The dominant organism in this bloom was Lactocyra. Some people might probably realize it used to be called Melicyra. It's a Phytoplankton. It's basically a benthic form that gets suspended up in the water column and then stays active. And these kind of organisms have been studied in other big systems like the Great Lakes. But this kind of bloom is reassuring to see. It's the kind of thing that we want to see. We still need a lot of information on how well this gets transferred through the food web. So what happens to this production and how it gets into the food web is still, I think, a big unknown that we still need to some resources. There's also the harmful algal blooms that you've been hearing about. And I'm glad to see Peggy's in the audience. But again, 2016 has not been a good year for microcystis and some of the other toxins that we've seen. And one of the recommendations of this panel back in 2009 is study the toxins in more detail. We're getting more data on the toxins. However, I think a little caveat on this is that we do not have sustained long-term funding to monitor harmful algal blooms in the Delta. And I think that's something we really need to pursue. We need to know year in and year out that we can get this kind of information. So So worst blooms this past year were in the South Delta. There certainly are a number of water quality effects that are documented with these blooms, some low DO and some large swings in pH. Many other freshwaters in the Delta are not particularly well buffered. And we know that microcystis like some ponds around certainly show that. The other thing I wanted to comment on is that the Delta has, I think, is blessed with a lot of really high quality sensor data that's being generated within the U.S. Geological Survey has been a leader utilizing this kind of instrumentation within the Delta. So there's a lot of interesting data that's coming already, and there's a bunch more that's kind of being tested and beta tested right now. And I was talking to Brian Bergamaschi earlier today, and they're doing a pretty good job of now at least a couple locations of continuously measuring quality. Not an easy measurement to make. We're seeing progress on that kind of methodology. Here's some example of the kind of data that's now becoming available to us, and these are the kinds of data. The data in gray and black is the flows, and the black is the tidally average flows within the Delta for a little over a year time period. August of 2013 to October of 2014. You can see the big flow event. The big flow events are also the time when you get big pulses of nitrates. This is a strong correlation between flow and nitrate as it occurs, particularly in our Mediterranean-dominated climate, where a lot of these high loadings occur January, February, March. How do you use that data? Well, this is my own thinking here, and this is basically from work that we've done both in New Mexico and also that a colleague of mine at the University of Florida, Matt Cohen, has been doing. These are nice systems. They're springs, but you can utilize these kind of continuous sensor data, phosphate, for nitrate, or oxygen. You also add some temperature and some light information. You can estimate rates of primary production. You can estimate rates of total ecosystem respiration. And if you actually then look at some detail at the structure on a diurnal basis of the nitrogen and dissolved oxygen, you can utilize this information, the nitrate information, dissolved oxygen information, to look at the autotrophic and heterotrophic demand for the nitrate, the autotrophic assimilation being shown there in green. And if you plot gross primary production estimates using the diurnal oxygen curves against nitrogen assimilation rates using the information from the diurnal nitrate curve, assuming the nitrate is your dominant source of organic nitrogen utilized, you can come up with these kinds of relationships, which are on a daily time step. And basically you can get, for example, a molar carbon-nitrogen ratio. Carbon on one axis, nitrogen on the other axis. In this system in Florida, that ratio happened to be about 24 to 1. That's probably your carbon to nitrogen ratio for aquatic macrophytes, an aquatic macrophyte dominated system. If this was an algal dominated system, that number would probably be down around 7. So basically you have some information you potentially can utilize to look at who the front producers are within a system. Finally, the wastewater treatment plant is going to be upgraded in 2021. It's a big, large-scale experiment. It's an exciting experiment. It's one that's been monitored well in some other systems around the world. The best that I'm aware of was the upgrades that were done in Brisbane, Australia, associated with the Healthy Waterways Initiative. This upgrade of a plant, they basically used the aquatic macrophytes in the system to actually look at the signal that was effluent dominated. A heavier N15 signal, and the series of diagrams you see across the bottom here basically is looking at before the upgrade occurred, as partial upgrade occurred, as complete upgrade occurred, and basically you can see that the reddish area, the area that is indicated high amount of effluent nitrogen in those plant tissues, was removed by this plant upgrade. This was a tool that they used to trace those changes through time, with nutrient loading to Moreton Bay being one of the issues that they were trying to improve conditions because they had a eutrophication problem in Moreton Bay. Okay, future nutrient studies in the Delta. This is my wish list. I think the sensors, primary production, respiration, nutrient uptake data, we've really got the sensors already. Let's start utilizing that data to do some of these calculations, see if they work. We've heard this over and over again today, and I think it's definitely a right-on pathway forward. We need to link hydrodynamics and nutrient chemistry. To do that, we need rates. We need well-measured rates of a lot of the key processes in the nitrogen cycle. We need to have them measured spatially and temporally. So that's an important part of making that work. I think the nutrient dynamics assessed in multiple aquatic habitats is an issue that the panel brought up last time they were here, and I would resonate that we've got all of these submerged aquatic macrophytes, we've got all these beds of floating aquatic macrophytes. We need to do more work with those kinds of systems. And then finally, the stable isotope tools. I think there's actually quite a bit going on in this. It's been focusing mostly in the open water, but I'd love to see more of this kind of stable isotope work being done. Some of the sloughs, the backwaters, and some of the macrophyte bodies. And then a little ending to say, okay, yes, there is a review article that Rai Bergamashi, Alex Parker, Maggie Chrisman, Annie Adelson, and I have written. It'll be coming out in San Francisco Estuary and Watershed Science in December of 2016, which hopefully provides a good overview of some of the nutrient issues within the Delta. With that, I'll be happy to take questions.
Yeah, let's take some questions for Cliff, and then we'll bring all the speakers up and have a more open discussion. So if there are any questions specifically for Cliff, go ahead.
The sensor technology seems to have a lot of promise, and you touched on it in your talk, particularly for illuminating more thoroughly system-wide metabolic processes where there are hotspots, where they're not. Could you give a little more information on where investigations stand right now in the system in terms of the network of sensors and what the information is being used for right now, and then sort of what it would take to enhance that to get to the level that you suggested in your future research?
Okay, a few things. First of all, there's probably people here that can jump up and make sure that I'm reasonably accurate what I'm going to say. There are about 40 locations within the Delta where they have SON systems that are being telemetered. And I think the SONs almost all have turbidity associated with them, in addition to chlorophyll on most. So there's good network of just basic SONs. There's about 8 locations where they're doing continuous nitrate measurements. How many of those have got total phosphate or got phosphate on? Okay, so phosphate and ammonium are an intermittent basis, but the total, the Cycle P phosphate analyzer that's been developed in the oceanographic world works well. We've been using a lot in New Mexico. We're very happy with it. Turbidity is its biggest problem. We have to make sure that the water you bring in to the into the measuring cell is void of any kind of turbidity. So you have to have good filtration basically on the front end to make it work. So those kinds of, some of the other sensors, for example, some of the dissolved organic matter sensors, some of the fluorescent dissolved organic matter are routinely being measured at some of these sites. The key issue to utilizing some of this technology, the one variable that you need to know well if you're going to do things like rates of primary production and rates of community respirations, you need to know something about the exchange process between the atmosphere and the water column. So you basically need to know the pre-aeration coefficient. There are methods to do this, for example, using trace gases like sulfur hexafluoride, but there are also techniques that utilize aspects of using modeling runs to actually make the estimate, kind of basically back calculate out the pre-aeration. We haven't really gotten too much into calibrating some of the estimates here in the Delta, but I think that's a pathway that there's interest in pursuing. So hopefully we'll make progress. We certainly have the database to work from. If we had more stations that actually had phosphate, ammonia, and nitrate, then we could do some of the ratio information you're looking at. We could do more of the rate calculations. That's definitely we can get at the community respiration. Does that cover what you were interested in?
Right. Could, in terms of where the SONs are now, or the sensors are now? What drove the decision making in terms of where they're located?
I believe they're all co-located where there is discharge information. So they're always located where there was places where they had good discharge information. Other selection criteria that you utilized, I suspect in many cases it was because there was interest in things like what the Liberty Island Cache Slough complex was delivering to the downstream areas. They were interested in some of the ideas of upstream subsidies affecting the food web downstream, and those kind of questions were also, I think, part of the decision making. I'm not part of the network, so if anybody else would like to give more information on site selection, but 40 locations for sort of some of your basic flow and water quality parameters, another 8 where nitrate is being utilized or measured, and a few select sites for some of these other sensors deployed and tested.
Yes, do you do any vertical profiles? It seems like this is important with Lisa and Jan's suggestion that the diatoms sink out on neap tides, for example. So do you have any profiles that could back up that suggestion?
As far as I know, none of these instruments are vertical profiling instruments, but some of the ones off the boats, I believe, that they deploy can be utilized to get vertical, but I don't believe they have moored set locations, nor are they using some of the...
So there are some profiles then, is that right? But
vertical, yeah. I'm getting a yes from some of the people who actually make the measurements.
And are they in connection with spring and neap tides?
Yes, I mean, that's a very important part of filtering out the flow dynamics is the spring versus the neap conditions. And yeah, we definitely see some pretty strong changes, much as was suggested and much as you're questioning earlier. I think it's a very important thing to factor into the analysis.
Do you have any current speeds on spring versus neap?
I don't personally.
Not
I suspect I could link you to somebody who does.
A relative amount. Is it a small difference or a large difference? Or
don't know.
Cliff, could you go back to your wish list slide there? I think the second one, the linking hydrodynamics and nutrient biogeochemistry, is really key here, particularly since, based on what we've heard and where the priorities seem to lie. And I'm just wondering what your thought is in terms of modeling and getting the modelers. It seems like there's a lot of good models, or there are a lot of modeling efforts going on right now. For example, the Lisa Lucas's models dealing with the linkage of hydrodynamics to grazing. I'm just curious what your thought is on linking those with sort of more biogeochemical questions, because it seems like really that's that's kind of the nexus, I think.
Yeah, I mean, I completely agree. In fact, you've got people in the audience that are really, I think, actively trying to make this a reality. We actually have some very good hydrodynamic models of the Delta, and many of them have gone from one-dimensional to two-dimensional to three-dimensional. You're here sitting in the front row is a person who can tell you a lot about the modeling effort on the hydrodynamic side. Getting the biogeochemical couples to those models is going to require good rate information, and in some cases, still looking for some key rate terms, but there's been a number of recent workshops that have basically come out with exactly the same recommendations. So I think next key is to get the resources to get people who do different types of models together in the same place, talking about how to couple some of these different types of models so that information from one model can help drive another model, and we can actually also intercomparison between models, and we can also see how sensitive some of these models are to utilizing different rates that are measured or estimated from other systems.
Yeah, I think the models will also be able to tell you some of the informational gaps, some of the, including some of the conceptual gaps that we may not be thinking about in this context as well. And then the other question I had was on stable isotopes. what are you thinking in terms of types of applications of stable isotopes in this rather complex system?
Yeah, there's a number of different types of studies that have been done using stable isotopes. I used it in a generic term here. I mean, most of what you find in the literature is nitrogen and carbon type of stable isotopes. But we did commission a review article here recently that Carol Kendall and Todd Miller have written for us, looking at the full suite of different types of stable isotopes that could be considered. And although there are a number of other isotopes that don't often get as much employment, like boron, for example, that might actually be very helpful with understanding some of the hydrodynamics in some of these systems and water sources. I think we've left it pretty broad and pretty vague at this point in time. We have a work group that's kind of pursuing what might be some pilot studies that we could try to do. I'd like to see by 2017 we could actually begin to generate some of these kind of data. 2021 is coming fast, and there's going to be a significant change in form and the amount of nutrient that's being put into that part of the North Delta. It'd be nice to actually get some of these kind of measurements before the upgrade occurs, and so we can get some pilot studies. They prove fruitful. They've given us the kind of information we hope that they're going to give, and I'd like to try to develop a long-term sustained program that actually does this routinely year in and year out and follows through the changes that are going to occur in the next 5 years.
Back to the models, will you need a subset of these hydrodynamic models? The system is spatially very variable, so what are you thinking in that terms?
There are a number of models. Bill Fleener, I'm looking at you. There are quite a few models that are, I mean, of course, which model you're going to use is depending on which question you're asking, but there has been a lot of improvement in some of the three-dimensional modeling capacity. The Deltares model, it's the model out of Stanford. Anyway, I'm not a modeler. I have basically been facilitating trying to get some of these modelers that do some of the physical hydrodynamic modeling together. We'd like to have them do some intercomparison using the same databases and actually look at some of the outputs from the models, much we work with GCMs, do ensembles of models that get the same kind of ensemble approach. We probably work in trying to look at some of the model outputs here in the Delta. But depending on where you are and depending on the complexity, for example, that many of the areas where there are confluences of multiple channels coming together, you need that three-dimensional capacity. In other areas, two or one-dimensional model they will work just fine and will be a lot less computational.
Do you have a data center here then that there's a wide range of people working in wide range of topics that they have to deposit their data after a few years? And
interesting that you should ask. There is a brand new law signed by the governor on the 23rd of September requiring that by 2019. So yes, we are in a very active stage of talking about how best to make our data available in a format that is usable by a wide variety of folks. And they have about 3 agencies that will be leading this effort: in the Department of Water Resources, the California Department of Fish and Wildlife, and State Water Resources Control Board. So those 3 organizations are going to be charged with basically making this law a reality, and they have about a 3-year time frame.
And are they going to go back in time to gather up the data in the 60s and 70s and 80s and
I don't know the answer to that.
Rescue it?
I don't know the answer to that. I mean, we have a couple data sources that you can go to, one run by the Department of Water Resources, the other run by the USGS. So there are places where you can get good data, but it isn't all-inclusive, and some of the old data, like you say, is still in cabinets or still in somebody's office, and so there's certainly more to be done on that, but it takes resources.
Any other questions for Cliff? Great. So what we'd like to do is have any of the other speakers who are still here come up and populate the chairs at the front of the room. So give the panel the chance to ask you guys some questions covering anything that was brought up today. And then after the panel's had a chance, then we'll open the floor up for comments and questions from the audience. So hopefully you still have some memory of what happened this morning.
Yeah, right.
Or at least some notes.
Checking my notes here.
Halfway
See what-
legible, right.
I have a question for Janice. Can you refresh my memory? Will we have access to the panel or the speakers tomorrow if we have any questions?
I'm just going to grab this one. Depending on your questions and their availability. Some people know that, well, Bill and John and Dave in particular have committed to being available tomorrow. If you have specific questions for other speakers or on other topics, let me know and we'll see what we can do to, if they're not available tomorrow to chat, maybe we'll have to do it some other time.
I have a general question for the panel. We've talked a lot about nitrogen and carbon, and I was just wondering if anyone, well, I'm just wondering if phosphorus is an issue in the context of changes that are going on, particularly maybe changes that don't necessarily involve the wastewater treatment plant, but changes in agricultural practices in California. And the reason I bring this up is because, interestingly enough, in other parts of the world, other parts of the country, some changes in agriculture have led to a renewed phosphorus issue, including animal waste, for example, chickens. I don't want to just bash the chickens, so let's put pigs in there too. Other issues, should we worry, is there any reason to be concerned about phosphorus, aside from having sensors to measure it?
Sure. Take a whack at it. So, I mean, there's not much of a signal for phosphate in the northern delta. There is, except, there are certain times, like, as I pointed out in the ship channel at the very top, where they run out of phosphate. For the most part, the story is about nitrogen. On the San Joaquin side, they have very high nitrate compared to the North Delta, and occasionally there, phosphate will run out. I know that, ag practices tend to, they're focused on reducing sediment loading, so that reduces phosphorus loading. So I think there's, still a lot of effort devoted to that. That's all I can think of.
I guess one reason for asking this question is that I'm wondering if like you mentioned, some increased treatments for phosphorus is actually exacerbating the nitrogen limitation or nitrogen impact issue. And I don't know how much phosphorus storage there is in the lower bay and how much imported phosphorus there is from the ocean to balance any of that off. But I'm just throwing this question out there just so that if 10 years from now someone comes and says, well, why didn't you guys work on phosphorus in addition to nitrogen? Why did that happen? But maybe there isn't an issue. I'm just wondering. For example, in North Carolina, we're starting to get a issue with phosphorus again, even though we went through all the steps in the 60s and cetera, with phosphate detergent bans and all that other stuff, but agricultural changes have really led to major changes in phosphorus releases, particularly episodic ones. So I'm just curious about that. But if it's not an issue, that's okay.
Hans, I'll posit a hypothesis, and that is that one of the things that we may see in California, like we've seen in other parts of the western United States, is that the parent geology makes a big difference. And so in the northern part of California, you've got a lot of saltic and fresh volcanics, and so there you've got phosphorus-rich, nitrogen poor source. But then when you get into other areas, like in Sierra Nevada and other parts of the state, phosphorus could be playing an important role, particularly in some of those streams and rivers. I just don't know what data exists on that, but I would start with the lithology myself.
Okay, yeah.
Okay. In terms of the, I'm sorry, go ahead.
I just want to say that I always think it's surprising. So we do collect phosphorus data, and we look at it, but mainly we kind of don't think it's that important because we don't have eutrophic conditions, which makes, usually you're looking at, oh, maybe we have too much phosphorus that's stimulating phytoplankton blooms. I think it's definitely worth looking at phosphorus and NP ratios and see if that has something to do with HAB blooms. And one question that we did ask Regional San is whether the wastewater treatment plant upgrade is going to affect their phosphate output. So they do put out phosphate. It is an important source of phosphate to at least the northern delta. I'm not sure what other sources of phosphate. And they do not anticipate their phosphate concentrations to change. But I think that's, something that we'll have to also measure. It will be interesting if there is some change in phosphate loading to the system following the wastewater treatment plant upgrade.
Yeah, that was my other question, actually, about the upgrade.
Their engineers have told Tim, who has told me, that they don't expect to see a significant change in their phosphate concentration. Okay.
I'm usually on the other side of that coin. In fact, been accused of being on the nitrogen mafia lately. I think. Just wanted to throw it out there.
One of the things we need to do is to, in our report, is to earmark some of the knowledge gaps. And we've been making notes and we've been reading these many reports and long reports. And I was talking to Dave and I asked him a couple of questions. He said, well, that's not on my top 10 list. So perhaps we could just go along the row here with all of the speakers and maybe give me what you think is your top priority from your area or your viewpoint as to what the knowledge gap would be, which then would translate into some future studies and recommendations. So just in case we have missed something, it'd be nice to refresh our memories here as to, from your standpoint, of what you think is really important to do. So maybe starting at the end there. Cliff has his list.
I have
already.
mine up, so I'm going to pass.
Yeah, good.
Well, I also had a list in my talk that you can peruse at your pleasure. But if I have to pick one right now, I want to reiterate the need that we do need these models, these biogeochemical, hydrologically linked models, but I think that the modelers really need values to put into those models. And I think that's a challenge. Our group has been interested in trying to look at nitrification rates as well as ecosystem nitrate uptake rates. We don't know much about sinks of nutrients to aquatic vegetation, either the invasive or the wetland. So I think that's an area that we need to, at the same time that we're trying to build these models, we have to create the data that will make them tell us something real.
And those rates could be lab estimated and then on probably into the field. Is that right?
That's correct. So, since I'm sitting next to Jan, I'll say, we need accurate clam grazing rates.
Right.
We have these huge ranges, so we can run scenarios, but we need rates for things like denitrification, nitrification, uptake,
right.
recycling,
of nutrients, recycling of phosphate, things like that.
It's too far off track, but I assume that that would mean also maybe putting more focus on other forms of nitrogen than nitrate and ammonia, such as solid organic nitrogen, all of the forms that factor in total nitrogen are getting into rates.
That's right, including the benthic sources. And if you look at the work that Davidson did, they have their box models when they say, well, where did that 30% of nitrogen go? Is it stored? Is it lost to the atmosphere? Or maybe it's 25%, whatever. That's lot. The delta is a sinkhole.
It seems like quite right now, quite a lot of effort is on the water column, the pelagic area, but the benthos is really important here, and you have some of these quite shallow areas here. So where are we in terms of that kind of information that would definitely need to be fed into, say, shallow water model?
I am aware of very little work that's been done in that area, but I don't know if anyone else in the room.
Okay, let's move on to the next. What's your biggest data gap?
Well, I'll answer that question first, and that is we had somebody in our lab that did nutrient fluxes in North Bay in the shallow water, and that is available in the report. It was a 1 or 2 time deal just to see what it looked like. He routinely works in Klamath Lake, so he's pretty experienced at this. And he does connect it to benthic organisms, which is one of the common fallacies in these studies, is to assume that nothing is mixing. And he does connect it to that. I think that being in that benthic group that everybody would like to have more information on, number one, there's not very many people who work in that, but if we could make sure, and living in the world between modeling and the data, if we could make sure when we collect certain data, other data is always collected so we can calibrate the models, that would be, it sounds very basic, but Hans Shaky said it's not basic. Collecting grazing rates without collecting current chlorophyll numbers, I mean, ideally, the whole thing, give me light attenuation, give me all the things that I would need to be able to calibrate a model would be wonderful. And the people don't do it because it's expensive, but they also don't do it because they don't think about it. Oh. So in some degree, it's a matter of just getting people to realize, yes, there are people who would be willing to do that. That you're doing that at that moment. You're running an experiment, please give me a call. I'll see if I can figure out how to do some different things.
So following up with that, how much interaction is there? So if somebody has some money and they're going out and they're doing these series of experiments, who actually knows about it and what they're doing, and do they get feedback from others, or what?
I think it's gotten a whole lot better in recent years, certainly in my career.
Okay.
I work with pretty much the people in the USGS know what's going on. I also work with Irwin. I work very closely with the Department of Water Resources. I try never to take a sample that somebody else has taken. So I really try to utilize what they already have and reanalyze the samples in a different way. So I think it's better, but it's hard. It's really hard. Cliff is in a position of trying to get us organized periodically, and a lot of sympathy.
Yeah.
Do you list, or do you have a predetermined schedule in terms of when the U.S., for example, when the USGS monitoring run happens on the up the river?
For benthos?
For anything. Do people know what the schedule is of the other group?
Right.
Oh, I think the USGS schedule is really well known. It's on the it's on the web.
So it's just depending on if there's a weather issue or something like that would be. Right.
And in recent years, we're combining the benthic cruises onto that schedule on the web so people know. But we don't do the kind of sweeps that they can do in water quality, obviously.
Yeah.
Okay, Liz?
So the USGS is sort of the headquarters for disseminating interactions. Is that right?
Oh, I don't know that I would say that. I think it depends what you're. I don't. I don't even know how to answer that. I guess the Interagency Ecological Program, the IEP program, is trying to wrangle cats. Does a pretty good job actually of getting people in the room once a month to know who's doing what. It's just there's a lot of stuff going on.
Yeah, I know exactly. That's why I asked.
It's even for those of us who've been here a long time and know a lot of people. I'm surprised at every meeting I go to. If something is happening, I had no idea.
I think, I don't want to take up time today, but since you have David Sen at your disposal, maybe tomorrow you can ask him about the Delta Regional Monitoring Program.
Right, that's another good example of an effort to do that.
Because I think that that's one issue that came up when the Regional Monitoring Program came in. It was, again, people brought up the challenge of interconnected data sets and coordination and it does take money to coordinate between different agencies. So there are definitely different sampling programs. High frequency stations are run by different agencies, and yeah, ask David Sen more about that.
Liz, what's your top data gap?
I think narrowing the gap between what is done in controlled environments and field experiments. These landscape scale experiments are complicated and they introduce a lot of variables, but if there's some way to utilize these Eulerian measurements we have, that, as Cliff mentioned, all those fixed station measurements are abundant. With my group's high-speed mapping capabilities, we're able to get out there more frequently and see seasonal variability in between where stations actually exist. And coupling that with some of the things Erwin and Cliff brought up about these landscape scale nutrient studies, particularly with isotope tracers or something, would be really useful.
Great. Yeah, go ahead.
Even though I've always been more of a field scientist, I've fallen the camp this time of thinking that probably the biggest gap is on the ability to grab a model and use it in a complex system like this to try to stitch together, weave together all the intricate parts and examine hypotheses. And if I was forced to make a choice between going out and collecting additional data to calibrate a model, especially as was mentioned earlier, that hydrodynamic models there's very good hydrodynamic models for Delta. There isn't really any biochemical models. They're well developed across the full suite of parameters and processes. So my guess is that the spatial and temporal variability in processes like denitrification, that they're going to be very spotty or very heterogeneous. And I would imagine that it would be really valuable, as Hans said earlier, to use models as a sensitivity analysis approach and identify data that's been developed in other systems, corrected for temperature, and adjusting those for this system and seeing how much we can explain or twisting them as hard as we can to see what the sensitivity is to the outcome.
Go ahead.
So I can only speak with respect to phytoplankton and phytoplankton physiology, where I think the data gap is. So one of the things that intrigued me about my own data is the huge differences in the FURFM with seasons and also with geographic locations. And I'm really curious, and I wonder what drives those differences. And so one thing that I can think of is irradiance. For example, so it would be neat to know how does light limitation really drive productivity in the system, and differences in productivity and chlorophyll accumulation versus the other factors. But even more than that, right now I'm really curious to know how does toxicity from pesticides and herbicides, and there seems to be so much in the delta and so many different compounds, and we just have no idea. That would go to the top of my list. And actually Stephanie Fong touched on that. To me, that data gap is really important to fill, to do controlled experiments with phytoplankton to see at what levels does it affect them, the different compounds that are floating around.
And that could be done pretty cheaply with this FV over FM as a first order screen.
Sure. But yeah, and it could be done cheaply with the regular 96-hour, toxicity assays that the EPA, all combinations thereof. Sure. Yeah.
But we probably need to extend that on into the invertebrates, right? Which might even be more sensitive than the phytoplankton.
Yeah. I mean, I don't know.
Okay.
So I was going to say rates, and then rates was said, and then I was going to say the benthos, and then benthos was said. I want to echo what Dave said about the need for biogeochemical models that we can use, and I just want to draw a linkage there to all of these great sensor-based data sets that we're accumulating, which are not just about calibrating models, but all of these data are also about validating models that we then build, and it's just important to flag that. I was going to say macrophytes, but that's been said several times. I was going to say pesticides and herbicides, but that's been said now. And so part of what I want to emphasize is the macro scale environmental changes in this system that are going to influence whether all of these rates and other things matter to anything we care about. And I think some of that comes down to understanding flow regimes, the impacts of climate change, and the changing bite environment. And I think it's important to understand that. We do have a lot of information about that, but what we need to do is link that information with all of the other information and all the other data gaps as they get filled in. And I can't escape going back to Dave's original schematic, because fundamentally all of these hypotheses lead to a set of things. And those things, which he didn't use this term, but I'm going to use it, those are ecosystem services from the Delta, right? And to what extent all of these things matter to changes in those ecosystem services, that's why we're all here today. And so we can't not include those in the hypotheses that we investigate, those final linkages. We need to study those hypotheses, and we need the models, and we need the benthos, and we need the light, and we need the macrophytes, and we need the herbicides, pesticides, and we need all the rates. We need all of that for those hypotheses, but where they flow after that is into the changes in the overall state of the ecosystem. And that's going to be driven by not just what happens in the Delta, but by larger scale factors as well.
Great. So we're going to take Erwin's suggestion, and then we'll have Janice with a microphone for anyone in the audience who has a question or comment.
Yeah, I would just agree with Cliff Dom. The big gap is the macrophytes, although it's 30% coverage after several multi-year droughts. So maybe if we get a good couple of flushing events, it'll become less of an issue. But it is a problem for the Central Valley Project, is that our pumps draw water towards our trash racks, and that stuff just accumulates and really raises havoc in operations sometimes. So I'd love to be able to solve that problem. I think modeling is really important, but the recommendation was made in 2009, and we still don't have a model, and there were recommendations before that. And I think we've made a huge amount of progress on the physical side. The hydrodynamic models that are used now are just orders of magnitude sophisticated than what, say, Totoro was using in the 1970s. But the biogeochemistry is pretty much just the same as what Totoro was using, kinetics, nutrients. That part hasn't really gotten very sophisticated. It's pretty much just disco-era technology in that sense. So really, I think the gaps are trying to validate the models not just with the sensor data, but actually doing big-scale experiments. And that's how you validate the model. I would argue that most of what we call validation now is really sort of pseudo-validation. It's, you know, taking half the data set and calibrating with it and taking the other half and validating it. But you know, true validation is when you make some big change and have a calibrated model, you know, compared to what actually happened. So for me, the big gap is, you know, getting permission to do big scale experiments so that you can actually see how the system's really working.
Okay.
I have another one.
Go right ahead. Please.
Clams. Why aren't there any clams in the upper part of the ship channel? I mean, that's, I think, a really mysterious question. I'd love to
Thank you. Okay, there are a lot of people in the audience who've been waiting patiently all day to have a chance to interact with the panel, so please wait for Janice to get you the microphone. And please identify yourself for the recording. Thank you.
I have a few fingers, so I can take a couple of hands.
Well, my top two suggestions about...
Can you please identify yourself?
Oh, I'm Brian Bergamaschi. That was just the recent advice. I need the Brian Bergamaschi, USGS. My top two suggestions about data gaps have been mentioned, but I'd like to reemphasize them. We know very little about how shallow water interactions affect the pelagic system, and I think we need to study those processes and much more intensely. Another thing that I think we need to better understand, Jan highlighted a little bit, is the role of detritus in feeding the main stem estuary. And we rely on phytoplankton to do all the heavy lifting for our ecosystem down there, but a lockable supply of detritus has not been separately characterized. But I think the lowest hanging fruit, and the reason I stood up, is one of the things that I think we need to do is we need to make sure we're all making measurements that are comparable, and we don't do a good job at that in this system yet. We need to have a robust, funded intercalibration training program so we can collect data that is comparable across agencies and across time.
This is obvious, but I'm going to mention it anyway, that in a lot of the documentation, I didn't see temperature mentioned very often. And really what I see in microcystis is that it's driven by changes in temperature. And our system has increased quite a bit, 2 to 3 degrees at least, the last time I measured it, from using continuous data. So it really makes a huge impact on the system, what species will grow, and how nutrients are utilized. And I think we really need to focus on how that is impacting the system and how that will impact the system in the future. Because what we decide is happening today won't be what's going to happen tomorrow.
Might just add something, if it's possible. Yeah, I just want to second Peggy's, that was Peggy Lehman, by the way. I just wanted to emphasize her point about temperature, but there's also kind of a synergy there with temperature and more extreme events like longer droughts, which, you know, if I were microcystis, that would be my ultimate wish, is get the nutrients into a system, get it cooking, and then slow down and then increase the residence time. So, you know, and all that's happening with the climate changes that we're seeing, not only in California, but everywhere, really, at least in the temperate zones.
Anyone else? Yeah, anyone else? Comment or question or recommendation? That's okay. I've been sitting all day.
Hi, I'm Linda Smith. I'm with the Metropolitan Water District interested stakeholder. I just had a couple of quick comments. First, today you largely heard, and a lot of your reading list is about work that was done in the North Delta. And there's, you know, good reasons for that. But the Delta is a big place. It's a very complex place. The picture of nutrients and phytoplankton abundance and composition is very complex, and it's different in different parts of the Delta. And so I know you don't have a lot of your reading list that is addressing things in other parts of the Delta. You do have a SFEI report that gives you some basic background data. But I would encourage you to think about that in your recommendations in terms of, studies and monitoring, that we're not just thinking solely about the North Delta, but we're thinking about parts of the Delta that are under high influence of the San Joaquin River, for example. And then also on a related note, and there is a sort of a separate stakeholder effort addressing this. As you know, with the water projects that we have, water is diverted out of the South Delta into both the Central Valley Project and State Water Project. And of course, those facilities are very different physically from the Delta, and we definitely see eutrophic conditions in those conveyance facilities and reservoirs. Some of the microcystis and toxin data that you saw earlier, that's happening in some of the reservoirs that are part of those systems. So that's another way where the nutrient issues in the Delta are connected to other areas that are very important. And then just two more quick things. There were a few comments and questions regarding monitoring, like when different people were presenting, you would ask a question about like how a sample was collected. For example, we often hear that in this system, we're very data rich. We've been collecting data for a long time, and we have wonderful data sets to analyze. But I think there's a lot that maybe your panel could provide recommendations on how we can be collecting some of that data better. For example, Dr. Pearl, you mentioned, how was the phytoplankton data? Was it a depth integrated sample, or was it just going down 1 meter? Or where can we really think about improving our monitoring so that we're getting much better information in the future? And then my final comment is, obviously today you didn't have any presentations from a lot of the Delta nutrient-related research that is covered by your reading material. And I would just urge you to obviously review that material and to contact those researchers directly. I'm sure many of them you know, that Dr. Dugdale and his collaborators and Dr. Corbett and her collaborators. And I would just encourage you to follow up with them as you're working on your white paper. Thank you.
Not to worry about that last point, but I'm curious, does every data monitoring program have a metadata base associated with it that actually describes, how the data is collected and the nature of the way in which it's analyzed? That's really critical.
Kind of depends on the program, Hans. It kind of depends on the program. Some are better than others.
I think it's really absolutely essential.
I agree.
Comment, and that'd be good to include in your recommendations, but we are actively working on that now. That's a huge effort in IEP.
So who's coordinating that? I mean, how will we ensure that it gets done?
Well, Jerry Brown kind of helped out with that.
Any more questions or comments from the audience? Okay, thanks for hanging in there. The last thing that we would want to let you know is that tomorrow afternoon from 2:45 till around 4, if it goes that long, the panel's going to come back and, for whoever is in the room, give you a brief update on their progress and where they stand and what the schedule might be for putting out the report that they're producing when they go away. And also, as you've heard a comment, the panel does have the opportunity to contact these speakers and other researchers while they're working on the white paper. So this isn't necessarily the end of their interactions with the research community in the Delta. Okay, Jess.
And it's not the end of the chance to ask questions. If you go home and think of something, feel free to email me, and then we can get that into the conversation. So, thank you. Yes.
That was a good way to handle it. What's your top for her to, yeah, just as a review for us, plus everybody.
So I have someone's thumb drive
Yeah, otherwise.
in the computer.
Yeah.